right, people, a very good evening once again to this amazing session and to your own channel, Unacademy NEET English. Yeah, we start again. Starting again is very good. I am Dr. Pankhuri Miglani, your biology educator. Yeah, with a lot of will and lot of good wishes, we have restarted this session and there should be no problem with audio and video. Please confirm it once again in the chat box. Ramesh Yogi, Reshu, please confirm that everything is superb now. Everything is superb. The team has worked really hard in fixing the challenges that we face today. And we are really sorry, I'm extremely sorry from my side for the inconvenience and for the wait that you had to do. And I really appreciate how you guys were so patient in bearing with us. Yeah, after all, you guys are lovely. All good, happy, smiling. Yeah, so let's restart. I want your pens and papers to be ready. If due to the technical glitch you didn't know, there's something superb that Unacademy has done. Yeah, nobody else in the country has ever done this. This is the first time ever that Unacademy UG subscription is absolutely free. But, there's a but, it is 5th to 7th of December. Okay, so please explore this. You can use my code PM1 Live for Avengers 3.0 batch, which is for NEET 2024. You have all the esteemed educators, your favorite educators. Yeah, classes are going on. I'm doing ecology in this batch. The classes happen regularly at 8 p.m. We are almost done with organism and population. It's a super batch, one of the best batches. And I hope to see all of you there. Don't forget to use the code PM1 Live. Also, the PYQ and the test series are free of cost again. Please explore all this and follow the profiles of all educators. Now we start with the mighty chapter of sexual reproduction in flowering plants. The day 3 of Game of NEET is going to be as superb, as exciting, as fabulous as day 1 and 2 were. Yeah, chemistry and physics were. Because this channel itself and the team itself is nothing short of being absolutely excellent. Yeah, and you guys are going to be excellent as well. Now, ladies and gentlemen, when we talk about sexual reproduction in flowering plants, we are talking about two major aspects. Yeah, the first one is sexual reproduction. And I want you to realize that sexual reproduction is a phenomenon which is biparental. Yeah, two parents come together, gametes are produced, gametes fuse together, and next generation is produced, zygote is produced. Now, also realize, be grounded of the fact that sexual reproduction is one event in the entire life cycle of any organism, and in this case we are talking about flowering plants. You know that flowering plants are the major hero of the story of plant kingdom. Plant kingdom, most advanced members are angiosperms. But do you remember from class 11th? Yeah, and I want you to remember. Guys, again, one request, please share this session with all your friends, family, everybody who was there in the session. Some people might not know that the session has restarted at 6 p.m. Kindly share this session and do good to others. It will be good for us also. Yeah. So now when I talk about angiosperms, okay, sorry, when I talk about plant kingdom, like I was saying, there are two things with which plant kingdom are defined as plants. You know, they are the diagnostic features. Number one, they appear green in color, they are photosynthetic, they have a similitude, they are autotrophic. And the second one is, there is something called as alternation of generation, which is not seen in us animal kingdom. Yeah, that is where we stopped. When 5 p.m. we met at 5 p.m. Okay, so we'll continue. Now, as the name suggests, I'm pretty sure most of you know this from class 11th plant kingdom. What is alternation of generation? Okay, now if you think about your own body, you and I, we are diploid organisms. Okay, and the only haploid phase which is very reduced is when we form gametes. It is negligible. But in plants, there are two very nice, very identifiable generations, the Sporophyte and the Gametophyte. Yeah, that is where the tune of botany starts playing. So, Sporophyte in plant life is the diploid or the 2N generation. We happy? Now, when this plant decides to reproduce, which is the cause of today's class, okay, we are going to get into the domain of reproduction today. Yes, yes. So, it has to do the reductional division of meiosis. Otherwise, I cannot form gametes. But in plants, I do not get gametes directly. That is how they are special. There is a full-fledged, appreciable sporophytic generation or the parent generation which on meiosis, and we are talking about life cycle of angiosperms particularly, forms spores. Now, if I am performing meiosis, the spores have to be haploid in nature. There's no two way about it. Now, a haploid organism cannot undergo meiosis again because N by 2 does not exist. Isn't it? N by 2 does not exist. So, what do I do? The spores will now develop into another generation or the haploid generation called as the gametophyte. Gametophyte. Boys and girls, sporophytic generation. Now this is a revision from plant kingdom. Mostly people just start studying this chapter with structure of flower. But if you understand what is sporophyte, what is ploidy, what is diploid, what is haploid, 50% of the questions that come from this chapter are anyway solved because they are based on ploidy. Okay. So now gametophyte is the haploid generation which is going to carry the gametes. Sporophytic is the diploid generation which forms spores. We are halfway through. So now this haploid generation, again, please don't get confused between meiosis and mitosis. As an N organism, I don't have the choice to undergo meiosis, Baba. So I am simply going to perform mitosis, and this is needless to mention, but I'm still making it clear. Mitosis, and quickly I'm going to form gametes. Oh yeah, I mean it. It does so quickly. Because in angiosperms also, you studied this in class 11th, gametophyte generation is very reduced. It's not here for long. The dominant generation is always the sporophyte. And that is why we call the life cycle as diplontic as well. Now, what is the fate of gametes whenever they are formed? They undergo fusion or syngamy. And now when N and N will fuse, gametes are always haploid in nature. There is no exception to that. There is no if and but to that. Gametes are essentially haploid entities. I am going to get the sporophyte finally from the zygote. The product of syngamy is zygote. Yeah. And zygote is going to develop into sporophyte, and that is how the life cycle happens. Now, today what we are going to study, what are we going to do today is what are the components of the class? Why are you here? You are going to study the structure of sporophyte with me. The structure of the parent. From class 11th morphology, you know that the parent which causes sexual reproduction is a very beautiful flower. But do all parts of flower take part in sexual reproduction? Let's see that. Let's revise that. After that, you are going to study the sporogenesis. Because we just saw, and that is why I reminded you and revised, why do you study sporogenesis rather than directly going into the concept? I am not talking about male or female as of now. Let's keep it simple. After sporogenesis, I know the fate of the spore, male or female, that is gametophyte. So when you study gametophyte, You study the structure. And what is the fate of the gametophyte? It is going to form gametes, na baba. So we study something called as gametogenesis. Now children, let's grow up and let's be very familiar with these terms. Some of you might already be good with these terms. So if this is scaring you a little bit here and there, genesis means formation. Sporo means spore. Phyte is plant. Gameto is gamete. Gameto is gamete. Genesis is formation of gamete. We are not concerned at all about the male and female as of now. And what is the fate of this gamete now? You tell me. It has to be fusion or fertilization. It has to be fusion or fertilization. But there is a problem. There is a problem. There is a topic which people miss between this. Okay, and that is not out of your syllabus. I am getting certain queries, certain messages. However good the gametes are, both the type of gametes in angiosperms are immotile. Female never travels, lower organisms or higher organisms. Female waits for the male gametes to reach. And in this case, male gametes are also immotile. Yeah. So a very important process that we are going to do today is pollination. Pollination. Ma'am, what is pollination? Well, today's class is special in a way. Even if you don't know anything about these terms, you are absolutely, this is your first biology class in life. I say even if a JEE aspirant, even if a mathematics student is sitting in the class, today's class is going to make sense for everybody, because I am going to deal with basics and then advanced concepts will happen invariably. That is the consequence when you are good with basics. Okay. So, if you don't know what is pollination, well, I am not going to give you a complex bookish definition as of now, but think about what needs to be done to bring them together, because fertilization is simply bringing the gametes together, isn't it? And what happens after fertilization? You guys know it. But zygote has to develop into embryo. So, a glamorous or fashionable term for the embryo formation in botany is called as embryogeny. Embryogeny. Now, somewhere somebody else might be calling it embryo development. But you guys are the Avengers. You are here. So, let's call it embryogeny. Yeah, formation of the embryo. There's no male, female here. With me on that? And then we talk about, once everything is said and done, there is seed and fruit. Now, seed and fruit is one aspect of this chapter which is done in morphology, and I am going to do it in morphology. When I take the class of morphology, which is the next lecture in Game of NEET series, we'll do seed and fruit in that. And lastly, what you have is apomixis and polyembryony. Okay, which is nothing but imperfect sexual reproduction. So when I say sporophytic generation, boys and girls, like I was saying, plants have a special organ which is called flower. But what exactly anatomically, if you look at this organ, what is it? Everybody in the chat box, what is a flower anatomically? It is a modified, or in better terms, condensed shoot. That's what the theory says. Now let's look at it practically. So if a shoot is like this, morphology tells us these are the nodes where the leaf arises. This is the leaf. Alternate phyllotaxy. Now, under the effect of hormones, when the plant decides, you know what, I'm going to reproduce now, instead of these leaves, what is going to happen? The floral organs are going to arise, which you have studied. For example, here sepals will arise. Or let's say here I'll become very pretty and become a petal. Okay, or here I will change into a bilobed anther. That is what they mean by a condensed shoot. But that's not all. The floral leaves, the leaves which were arising at the node have now become floral leaves, and this entire shoot is going to condense itself, and that is how you get the seat of the flower, which is the first portion, swollen portion of the flower. Okay, so this cylindrical structure is going to flatten itself, form a very nice seat. On that seat, the nodes, whatever were present, will now give rise to the floral organs. That is the meaning of a condensed shoot. Okay, so the first part, what are the parts of the flower? Simple revision, no problem at all in life. Yeah, this is just the revision of class 11, but very necessary. So the first seat, like I've been saying, is the thalamus. People call it receptacle, torus, let them say that. Yeah, we are good. On the thalamus, the first organ that will arise, the first organ is sepal. Now listen to my words carefully. I said the first part that arises is sepal, but it never arises singly. Seldom it does. We are talking generally. Okay. So the whorl of sepal you studied is called as calyx. We are going to focus on whorls today, and then the second whorl here is petal. Sorry, the second part is petal. Yeah, the colorful part, the attractive part, and this is called as the corolla. The corolla. Let's change sides now. This one here, what do you think is this? Well, this is the third part, which is the stamen. Listen to my words carefully. I said part, single unit, because that is also a question. Yeah. So stamen, and the whorl of which is called as the androecium. Androecium. Happy? Andro is male. Okay. And this part right here, let's mark it this side. This is pistil or carpel. Pistil or carpel, and together they form the whorl of which is called as gynoecium. To be very honest with you, you guys know it, that gynoecium doesn't shy away from being single. The female organ, the female part is always lesser in number as compared to the male parts. I think I'll focus on this when we have the morphology class. All good? Happy? No problem? So now what we're going to do is we are going to categorize these parts. If I talk about corolla and calyx, they are together called as the accessory whorls. And why? Why are they called as the accessory is the word? Because they do not take part in reproduction directly. Just like human reproductive system, you have primary sex organs and you have secondary sex organs. They are assisting. But if I talk about androecium and the gynoecium, they are called as the essential whorls. Essential whorls. Because my goodness, they are going to take very active part in the process of reproduction. Yeah. So now in this chapter, when somebody says you have to focus on the sporophytic generation, you focus on androecium and gynoecium. We just did this. What is the single unit of androecium? It is the stamen. It is the reproductive, male reproductive part. Now boys and girls, I want to make sure your basics are very good. Whenever the male part will be talked about, we use the prefix micro, okay? And I want you to think about, recapitulate the very fact at least of gymnosperms, if not pteridophytes, because they are the closest cousins of angiosperms. Yeah? Now, in gymnosperms, let's talk about gymnosperms, because that is where the confusion starts. And this is called as homology, not given in textbook, but it is the heart of the chapter. It is the basic tune on which the chapter flows. So in gymnosperms, God did not bless them with a special floral organ, the flower. Instead, some of the leaves stopped performing photosynthesis and became reproductive in nature, called as the sporophylls. Make sense to us? Yeah? Those leaves which become photosynthetic, which become reproductive. What do? If they become reproductive, they develop sac-like structures. Listen to the keyword, sac-like structure, and we name it sporangium. Are we good? I'm not talking about male and female. No. Let's focus on the basic terms first, so that your entire chapter goes smooth. And on sporangium, this sac is going to be filled with, or it is going to carry the spore. And rest you know that spores will develop the gametophyte generation. The entire life cycle you know. But let's focus on the sporophyll. If you think about the male generation of angiosperm, stamen is simply the microsporophyll. Because stamen is going to do all of these things. And you will just see it. You will realize it. Okay? So what are the two parts of the stamen? This is the second point is the parts of the stamen. Well, the swollen part of the stamen is anther. This is a very simple structure. It's rather the anterior structure. Yeah, it is the anterior structure. And there's a reason why I am talking about the orientation here. Yeah, and this one here, the stalk, is called as the filament. You know for a fact from class 11th again that this filament can be attached to the thalamus, cohesion, adhesion. It can be attached to a petal as well. Sometimes it can be attached to pistil also. You know all that. So now what I want to tell you, which is a very beautiful line of NCERT, the orientation as I was saying. This anther is rather away from the center of the organ. If you remember, in structure of flower, we are assessing everything based on the seat of the flower, which is thalamus. Baba, if you have to study the human body also, when you will dissect the human body in first year MBBS, the first class is going to be about anterior, posterior, antero-posterior, dorsal, ventral, lateral. Why? Because otherwise you cannot understand. For example, this is the midline, the assumed midline, okay, the midpoint, and this is close to the midpoint. This is away from the midpoint. Locomotion and skeletal structure, zoology. Similarly, filament is supposed to be proximally located. Very, very important. I'm not telling you anything special. It's given in NCERT. This is the first introductory line of anther. Proximally located. And this right here is distally located. And now you know why. If you observe this structure antero-posteriorly, what does that mean? If you are looking here, but you twist it, you turn it and look at the back side, then this filament is attached to the anther posteriorly. Anterior, posterior. Actually, it is ventral and dorsal. But just to make you understand. So, this filament, what we are going to write, is attached to anther through a tissue called as the connective. Are we good? We better be. So this is also the key term, connective, but it's going to repeat itself. Is that clear? Now, boys and girls, what is missing here? And sometimes you can't appreciate it if you look at the structure in a single view, that is anterior in this case. There is a groove or a depression. A groove is never a structure. It's a depression signifying something. In this case, it signifies one of the most important points, that anther, this anther is bilobed. It is a bilobed structure. Okay. So, once again, anther is a bilobed structure. It is distally located with respect to the flower. Remember, this is proximal, this is distal. Okay, take your own example, take the example of your own body and you will be good to go. And then comes the filament. Filament is proximally located and helps the anther to attach to thalamus or other parts. The filament is attached to the anther through a tissue which is sterile in nature. Very, very important to note. Connective is not going to take part in reproduction, hence we call it sterile, not fertile. Now what I am going to do is, I am going to take this scalpel, the same scalpel with which you will dissect human body in first year. You are going to take that scalpel and make a cut here, make a cut here. This is called as the transverse cut. Okay. If you are making a cut like this, it is a longitudinal cut. If you are making a cut like this, it's a transverse cut. When I make a transverse cut in the anther, because anther, ladies and gentlemen, is the fertile part, not the filament. Yeah. So, anther is the fertile part of stamen. It is the fertile part of the stamen. And if I open it up, what do I see? Wow, it's a nice structure. I want you to look at this structure and let the common sense grow up, rather than you trying to mug up the technical terms. If you look at this structure, oh yeah, these are the two lobes ma'am was talking about. I can see them clearly. Yeah, to make it more clear, this is lobe number 1, this is lobe number 2. Yeah, now I agree more that this is a bilobed structure. But I also see that every lobe has two sacs. And what do we call the sacs? We call them sporangia. Okay. So, total 4 sporangia are present in one anther. Okay. I'm going to make it a little more glamorous and call it tetra. Tetra is for sporangiate. See, that is why I told you the homologies and the basics. Now it's breezy and easy for you. So tetra sporangiate. Okay, ma'am, no problem. And because of these 4 sporangia, this structure has 4 sides to it. So I'm going to call it as a tetragonal structure. I have studied about the shapes, might as well call it. Okay? Now boys and girls, there is a term which is used rather liberally by people. But if I talk about precise and proper and correct botany, we shouldn't be so liberal. And I'll tell you the reason why. Let's get ready to know that. So for now, I'm going to write it in a different color. And I know that lobe number 1, keep listening and looking at the diagram, you don't have to look at me. Lobe number 1, lobe number 2. Lobe number 1, lobe number 2. Focus on lobe number 1. It has 2 empty cavities. Empty for now. Okay? And cavity in biology has a generic term called theca. Okay? So, dithecous. And that is how you see the features of anther. It's a bilobe structure, I know. It's tetrasporangiate, 4 pollen sacs. It is tetragonal in shape. It is dithecous. Now, the story doesn't end here. Let's go further. Well, the connective that I was talking about. The connective that I was talking about. Now you see it. It is going to contain the vascular strand. The very last class, the previous class that I took on this channel was about anatomy. And in that session, I spoke about the meaning, the bare meaning, nothing very technical or advanced, of vascular bundle or vascular strand in botany. It simply means xylem and phloem. Please don't keep them separately. They don't like to work separately in higher plants. Okay, so this connective right here, which we have mentioned earlier, no problem at all, is not going to take part in reproduction. It is rather going to contain the vascular strand, which simply means class 11th has taught us xylem and phloem. Okay, something more is remaining, and that is this constriction that you see here. Do you see that? Can you appreciate how the anther somehow in its outline makes a constriction here, makes a concavity, and that, ladies and gentlemen, is called as stomium. As simple as that. Now, stoma, even in animal kingdom, simply means plants, simply means mouth. Yeah, stoma is mouth. So, stomium here, or the constriction, simply is the mouth through which pollen grains will come out. When the anther is ready and mature, it is ready to send the next generation out. Go to the female, fertilize, you are ready. Okay, and that, my dear children, the release of pollen grains from the anther is called as dehiscence. So, the site of dehiscence, or some people like to call it line of dehiscence, is not random. Anther is not going to break wherever it wants, however it wants, whenever it wants. Stomium is the point where dehiscence will happen. Now, the story doesn't end here. as usual. And this is a very, very important question. Structure of anther. Every year questions are asked from this. Now I want you to focus on the diagram. Yeah, but what part of diagram? Focus on these structures. Yes, ma'am, these are empty sacs. We got it. They are sporangia. We call them pollen sacs. Yeah. Now, this is a little outside textbook, but it's interesting to know. Okay. So, one of the special points, if you don't want to know it, it's okay. But it's related to what is very important to know. There is a certain cell called as archisporial cell. And I am not going into the details, which forms these sacs. But the problem is, this cell causes periclinial divisions. Ma'am, what is periclinial division? My life. Last class, anatomy, I told you. When a cell is generous and decides to form a parallel stack of cells, both the sides. Okay? So, based on the orientation of the organ, you decide what is the outer side and what is the inner side. But this cell has claimed, I am not going to form cells only on one side. I am going to form cells on both the sides. When we do anatomy, I will speak more about it. So, this archisporial cell, boys and girls, what I am telling you will create a photogenic memory of the concepts. If this cell is present somewhere here, then on the outer side it is going to form these peripheral layers. And on the inner side or the other side for the time being, it is going to form a mass, a generous mass of homogeneous tissue called as the sporogenous tissue. Yeah, we will find the space and write it. Yeah, let's mark it again. And if you want to skip this part, it is okay. It is very much okay. You can skip it. But all you need to know is that an anther, the structure of an anther has the peripheral layers, which I am going to draw right now in some time, and it has the sporogenous tissue. As the name suggests, sporogenous tissue. That is, it has the ability to give rise to spores. We already spoke about the continuation. We already spoke about the life cycle that should happen, isn't it? So now what we are going to do is, we are going to take a microscopic view. Pick up your scalpels. We'll pick up our knives. You call them scalpel in surgery. You should, as a NEET aspirant, you should call them scalpels. So now I took the scalpel. I just cut the peripheral layer and some part of it, boys and girls, he's taking some part of the sporogenous tissue, but he's taking a bigger chunk of the peripheral layers. And what did he see? Well, he saw many layers. He saw a multi-layered structure. Yeah. And like is the case in any organ, whether it is anatomy of root or stem, the first essentially protective layer is always epidermis or simply the skin. Okay. So the very first layer that you encounter is epidermis. There's nothing to learn because this is present whenever you study any organ. This is always thin walled. I cannot have a thick skin now because I need only protection, but I also need the exchange of nutrients exchange of moisture. It is thin-walled parenchymatous, closely packed cells, closely packed, and if it is not mentioned otherwise in botany, assume that it is parenchymatous. Parenchymatous. With me? The primary and the only function in most of the cases. There are, of course, in science never use the word always, actually in life. Yeah, and I never thought that you guys won't be able to hear me at 5 p.m. Yeah, so that also happens. So thin-walled, closely packed parenchymatous cells, it is protective in function. Now the second layer, very good Mayuk, Gayatri, oxytocin, very good. Aren't you guys amazing? All good? Enjoying the session? Then when will you share the session? When is that going to happen? And I want a lot of fire and hearts. You know, Gopika ma'am made me used to this fire emoticon, but I don't remember my favorite. It keeps changing. Choose your favorite and send it. I want a Josh. I want a little bit of Josh. Manjushri, at least for today evening, please don't mention the word voice. It touches a nerve. And my darling or dearest, most of the people have the sound okay. I would really recommend that you check the speakers in your device. Thank you for all the love, guys. Thank you so much. Keep supporting us and keep putting fire to the fuel. Keep putting fuel to the fire. Yeah, a lot of fumbling happening today. Keep putting fuel to the fire, which is your love and support and appreciation and all the comments that you guys write. I read all of them, whether it's on the app or here. It's really, really lovely. Ruth, darling, since the concepts are so dense and we are going outside the textbook, we are trying to make the basics clear, I don't want to break the flow. That's the whole idea. And we got delayed by an hour. You know, you understand. Okay, interesting. Now, the next layer is a sweet layer. Very sweet layer. Doesn't trouble anybody. No problem at all in life. Do you see these cells? Again, a relatively thin layer, thicker than epidermis. Every layer is going to be a little thicker than epidermis. Okay. Now, this layer is special in a way, eyes on the screen, that the tangential walls have thread-like or fiber-like thickenings. Yeah, this is the speciality. And you know what? The examiner always frames a question out of what is peculiar and unique. Okay. So, we are going to mark this as endothecium. The sequence of layers is very, very important. Endothecium. What is the speciality? These fibers or threads that you see are actually made up of alpha cellulose. Alpha cellulose fibrous thickenings. Ma'am, why? Why God loves to create a complexity in the structure? Why can't the entire structure look like epidermis and just be more thickened? Why trouble us now, boys and girls? Somebody has to take the lead. When Anther wants to, remember we did that? Yeah, there it is. The stomium. When Anther wants to break open and release the next generation, which is ready to fertilize the female generation, okay, it requires assistance. This layer at the level of stomium, okay, creates a contraction owing to these fibrous thickenings. They are made up of alpha cellulose, okay, and they sort of pull the entire structure like a rubber band, like this, okay, and that is how the anther along with the epidermis will break open only at the level of stomium. I am not doing it on the other structure because I don't want infection or toxins coming in. Is that clear? So it helps in dehiscence of anther. These are the 2 points that you need to focus on. Next is a layer which is not unilayered, which is not a single layer. Till now, epidermis, single layer, endothecium, single layer. Now what we have is 1 and 2 and 3. If you are a class 12 student, examiner loves to give the question, please draw the diagram of structure of anther. You would need to draw this. This layer, boys and girls, the third layer rather, this is the first one, this is the second one, and the third layer is the middle layer. Middle layers, because they are multiple, but there is no stamping by the plants. They can be 1, 2, 3 in number. What is the function of middle layer? Well, you know what? It is short-lived. First of all, it's ephemeral or short-lived in nature. And for whatever time it is there in the life of an anther, it provides nutrition to whom? To epidermis? To endothecium? No, they don't need help. Well, you know that we are moving from outside to inside. Have a look at the complete structure. We are moving from outside to inside. This was the outside, epidermis, endothecium, middle layer. Here something important is present, na baba? Sporogenous tissue is present, who has a lot of task to form spores later on. Lot of developmental changes are going to take place. So it is going to nourish the developing structures or developing microspores. So nourishes spores. Okay, and now there is a lack of space. As usual, we are going to shift it. Short-lived and nourishment. Now, the last layer is the most important layer. Do you see? This is going to have very large cuboidal cells, and this is considered as the most important layer of anther. The feature is it will have a very glistening, very prominent nucleus. Very prominent nucleus, and it is the longest living layer. Understand, this is a previous year question, that this layer is closest to the sporogenous tissue. It's a direct previous year question. Which layer of anther is closest to the sporogenous tissue? And you guessed it right. It is tapetum. It is tapetum. So the fourth layer that we are going to mark here is nothing but the tapetum. Now tapetum needs a special mention because this layer is multi-talented. But I'm going to start from speaking about the primary function first. So we are going to write functions of tapetum. Now boys and girls, whatever I am going to write will not become clear to you in this very moment, because tapetum, like I said, assists the anther till the later stages which you are going to study with me. Gametophyte, gametophyte generation, gametes. It stays with anther till then. So till you understand those processes, you would not understand all the functions. But I am going to relate every function when the stage arises, when the stage comes. So like I said, we'll start with the primary function. Nourishment to developing pollen grains. Okay, then very dense cytoplasm. Binucleated or multinucleated or multinucleated. Why is this happening? Why do I have dense cytoplasm? What do you understand by dense cytoplasm? Whenever you observe a cell under the microscope and it has a lot of nutrients, carbohydrates, proteins, fats, it appears dense under the microscope. That is what I mean by dense cytoplasm. This is related to the very primary function, the nurturing function of tapetum. Okay. Other than that, what it is going to do is, it is going to form the Calais enzyme. Ma'am, what is Calais enzyme? We don't know. We would get to know. But we know for a fact that the word "ase" means something to do with hydrolysis. So yes, you guessed it right. If you guessed it, it's a hydrolytic enzyme. Good. Then it forms sporopollenin. Ma'am, what is sporopollenin? We don't know. No clue at all. Okay. But it releases special bodies, and one of my favorite questions, called as Ubish bodies. Ubish bodies, which are lipid rich bodies which helps in the formation of sporopollenin. Is that good? Is that good? Very good. I can see some of the wise comments, like it is the hardest organic material, etc. Then it forms something called as pollen kit. Well, as the name suggests, it's a kit which carries pollen grains. But this kit is seen only in entomophily. What is entomophily? We don't know. Okay, this much I'll tell you. Entomophily, insect pollinated plants. These are some very, very, very important points. Lastly, it is the job of tapetum to release certain compatibility proteins. On the surface of pollen grain, so that when pollen grain goes to the stigma, goes to the female and asks for acceptance, okay, the female will first look for these recognition factors or compatibility proteins. Then only the pollen grain will do the next set of events, or in other words, germinate at the level of stigma. So compatibility proteins Yes, don't you think Tapetum is multi-talented? Very good. Yes. Now, boys and girls, let's talk about the sporogenous tissue. We saw that here we have a mass of homogeneous tissue. What is the ploidy of sporogenous tissue? This is 2n. And we are calling this structure archisporial cell did its job. I told you, it formed the parietal layer and it formed a mass of cells which are diploid in origin. Now the plant is like, you know what, I've done everything for you. The parent is ready. Now you have to do something to get into the next generation. And the next generation, you know, what is Sporophyte going to do? It is going to form spores. We saw that in the beginning of the session. Any problem with that? Go back and see that. But we also saw that Sporophyte generation undergoes meiosis because let's face it, it has to get into the Gametophyte phase. That process, boys and girls, very, very simple. Since we are studying the male generation, we use the term male micro, and since we are going to form male spores, it is called as Microsporogenesis. Sporo is for spore, genesis is for formation. So if you have to define it, you really don't have to mug it up. It's right in front of you. The formation of microspores what is the product? Microspores from the sporogenous tissue. Now I've been saying tissue, okay, but meiosis has to happen in a particular cell. Now you will be surprised to know that all the cells that you see here, in case of male, even in human reproduction, millions of sperms are released for one egg. So almost all of these cells are going to become or develop pollen mother cell. Simply means that it is going to decide to undergo meiosis. So formation of microspores from pollen mother cell yes, or simply PMC by the process of meiosis. Simple. Now, cell division has always taught you that a single meiotic division always gives you 4 daughter cells. So, imagine that every PMC, and there are like thousands of PMC here, every PMC will form 4 daughter cells, and this way I get millions of not pollen grains but microspores. Promise me today that you will not say that the product of microsporogenesis is pollen grain. It is heartbreaking. Don't do that to yourself. Microspore is not pollen grain. I repeat, microspore is not pollen grain. So at the end of microsporogenesis, although yes, I agree, we're calling it PMC. Actually, there's a synonym. You can call it microspore mother cell. No problem at all. And then it becomes MMC. But I'm not getting pollen grain right away. Is that clear? So what is going to happen? Millions of microspores. This is one pollen sac. Very good. What is a microspore? Now I am going to make this confusion clear for everybody. Microspore is the first cell, okay, or precursor of gametophytic generation. Microspore is not pollen grain, microspore is not gamete. Please don't do that. Okay? The ploidy of microspore is N, and now the journey, the entire journey of microspore to pollen grain. Will start. But remember the basics here on. From here on, remember, I am already in the N generation. This cell is capable of doing nothing. From here on, you will remember your target or your destination. Your destination is to reach male gamete. You have just entered the area, entered the domain. That is N generation, the first cell. So if the examiner likes to ask, if he is like me, he would try to check the basics, and he wants to ask, which is the first cell, the first point, starting point of gametophytic generation in a male plant, in a male flower, then it is not pollen grain. I've seen people marking pollen grain. No. Pollen grain, let me break it for you, is the male gametophyte rather. And boys and girls, you have still not reached the destination. So, even pollen grain is capable of doing nothing. Even pollen grain is capable of doing nothing. If this is personally, I like to be precise, I use the word developed pollen grain, because if pollen grain decides not to germinate, I don't want to do it, you will still not consider it as a proper male gametophyte, because male gametophyte has to bear the male gametes. So, please clear this confusion. Pollen grain is not, everybody repeat after me, not male gamete. And your destination is to reach the male gamete, remember that. Good, everybody? So now we are going to go to the next stage, and what is going to happen? We are going to start from what we got. What do you have? You have the product of meiosis, a haploid cell. It has to cover the journey. Okay? Now this journey is covered in 2 stages. This journey is covered in 2 stages, boys and girls. Mostly. One is the pre-pollination, development, okay, and this is the post-pollination. Good? Happy? Yes? Who is this? Who is this? Everybody in the chat box. What is the another name of pollen grain? Everybody? Can you tell me? What is the other name of pollen grain? Wow, look at the, thank you. Rashmi, thank you for saving the day. Priya, very good. Ruth, very good. Excellent. It is male gametophyte. So this journey, boys and girls, this journey is called as development of male gametophyte. Can we call that rightfully? We don't have a problem. We don't have a problem because we ascertained that this is the male gametophyte. He's going to do something. We're expecting pollen grains to give us male gametes in the future. Remember, your destination is male gamete. So, pre-pollination means before it goes to the female generation, before it lands on the stigma, before that transfer in the parent's body is pre-pollination development, or in other words, precocious development. Precocious development, because it's happening within the anther. And this, in other words, is called as the stigmatic development, because it is going to happen on the female. I hope everybody knows. Stigma, style, ovary. This much basic of the female generation is good with us, right? Class 8th, 9th, stigma, style, ovary. Ultimately, this pollen grain lands here, asks for the female. Do you accept me? Compatible, it's a chemical dialogue. We'll talk about it, pollen pistil interaction. So, this is what happens. This is what we're going to study. So now when this happens, what you see here, I want you to ignore. I want you to ignore the wall that you see here, spiny wall that you see here, because it develops later. What I'm going to do is, I'm going to draw it here for you. How about that? So, in my classes, you will often see inclusion of NCERT diagrams. Of course, we'll draw the diagrams, the critical diagrams ourselves, but NCERT diagrams, because diagram based questions are asked in biology, more so in the new pattern, I'm going to include all the NCERT diagrams as far as possible. So that is just a cue for you to go back after the class and look at that diagram. Now this was the microspore. Poor guy just came into the world and he was told, you're not here for long. You have to develop into pollen grain. So what happens? It elongates like any dividing cell. And this guy has to go under mitosis. It cannot undergo meiosis because this guy is just N. It has a single set of chromosomes. Yeah. So now this nucleus and the cell enlarges, but something weird happens here. Something weird happens here, which is called as the vacuolation. Very huge food laden, food storing vacuoles develop in the cytoplasm of this microspore. Okay, with me? Vacuolation. Uh-oh. Vacuolation. The examiner is not really going to ask what is vacuolation, but he is going to give you the sequence of events. And if he says that the nucleus of the microspore divides after growing in size, then that is not true. The first step is appearance of vacuoles in the cytoplasm of microspore. What this does is, it pushes the nucleus to the periphery. Goodness, there's something wrong here. The nucleus, which should be in the center of the cell, is now pushed to the periphery. And the story doesn't start here. When it is pushed to the periphery, that is the time you will see the structure appearing, which we are not going to talk about now. We are going to talk about the cytoplasm only. We are going to focus on the cytoplasmic components. We are going to remain in this boundary. Whatever is happening inside, we are going to focus on that for some time. Make sense to us? So, nucleus push to periphery, and vacuolation happens. I am using the word vacuole again and again. Okay. Now what you see here, there is a longer spindle and there is a shorter spindle. Think about the metaphase of cell division. There is a necessity that the mitotic apparatus should be equal on both the sides. All of that story and song of aster bodies, organization of mitotic apparatus, the spindles are equal in size on both the sides when chromosomes align on the metaphase. But here I see unequal spindle or asymmetric spindle. Okay, so this is the first stage. Second stage, nucleus is pushed to the periphery, asymmetric spindle. Signifying nothing but one of the peculiar features, which is unequal division. Unequal division of this nucleus. This is still the nucleus of the microspore. Around this time, by the second stage, it starts looking like a pollen grain. But guys, I must inform you, these vacuoles have now fused together and I have a lot of food here. I have everything the plant can ask for. Carbohydrates, proteins, and unsaturated fatty acids. Now that's special. Okay. Now what happens is, owing to the unequal division, I'm going to speak about nutrients again. I'm not going to leave it at that. Owing to the unequal division, what I get is a larger tube cell. Repeat after me, larger tube cell and a smaller generative cell. Okay, we got somewhere. This, boys and girls, is called as the bi-cell stage. I'm not saying binucleated. Now these are some very precise concepts. Okay, the precision of concepts is important if you really want to ace your exam. Do you see that there is a wall formation around the generative nucleus, if you will? This wall that you see is callose. It is callose. Are we good here? We are good. So now, which is, I think, chemically, if you ask the chemistry of it, it could be some beta glucan or something. Not in your syllabus, just trying to remember. Tapetum is going to release the callase enzyme. Pollen grain or microspores have a close relationship with callase enzyme. In fact, the microspores which are released I told you 4 microspores are formed. Tetrad of microspores are formed. Initially, they are also stuck with this sticky substance called as callose. It is still in the anther. But before pollen grains will be leaving the home, leaving the parent, they have to be spread out, and that will be done by the callase enzyme. One point is sorted. One of the points is sorted, released by none other than tapetum. Okay. Now finally, as this structure bicelled, my heart doesn't want to call it pollen grain. Informally, you start calling it pollen grain because something is happening outside also. Okay. So this is the vegetative or tube cell. What are the diagnostic features? Large, irregular, very important point, irregular nucleus, very important point, food rich cytoplasm of carbohydrates, all the macros here, proteins, and this is the reason if I were the examiner I would give it as a AR question. Why would people decide to take pollen tablets so fondly? Because you know what? It has unsaturated fatty acids. Has anybody thought ever? Why unsaturated fatty acids? What are they doing? Because certainly plant has a lot of brain into its structures. Plant is not doing anything just randomly. It has reasons to it. Okay? Let me know if somebody knows why unsaturated fatty. So all in all, this is a very rich cytoplasm. Now let's talk about this guy here. This guy was called as the generative cell. You know why? Because this is the one, this guy is going to do all the hard work. What is the hard work? Your destination is male gametes, remember that. You haven't reached there. All of this is just the preparation, Baba. This guy, as the name suggests, you should get the hint, is small, spherical nucleus, no problem. Spherical nucleus, very thin layer of cytoplasm, not a very thick, enlarged, dense yes, dense. It has food reserves but much less than the vegetative cell. Okay, and more often than not, it forms this spindle shaped structure. Okay, so spindle shaped thin, dense cytoplasm. Of the angiosperms undergo dehiscence. Now, right now. What do I mean by now? It means when these two develop. The parent, the anther says you are ready, go on the stigma, and then comes the post-pollination development. Is that clear? So one mitotic division has happened and this is what I got. But in rest of the 40% boys and girls, what is going to happen now, or what we wrote earlier as the post-pollination or stigmatic development, that also happens in the parent. The pollen grain is very confident. Actually, in most of the monocots, that is the case. This is usually seen in primitive dicots. The monocot is confident that when I go to the stigmatic surface, it's okay. The couple will accept me. But here, the pollen grain is thinking, we'll see when I go there. If the female generation accepts me, then I will tell the generative cell. I will tell the generative cell to undergo the second mitotic division. Okay. So, before that, let's get to the point of dehiscence of anther. There's no surprise for you. There's nothing to tell you in dehiscence of anther. You guys know it, that endothecium brings about it at the level of stomium. But you know what? One of the keywords, before dehiscence or a mature anther always dehydrates to a large extent, so that it is easy for the layers, the endothecium, the epidermis, to shrivel away and to cause that contraction. Okay. So, dehydration or loses water of mature anther endothecial thickenings contract at level of stomium. At the level of stomium. Is that clear? Now, listen to me. There's one catch in the story. I like to create this catch. Remember when we were talking about the features of this guy here, anther? I used a different colored pen and I wrote the term diathecus. More often than not, by most of the authors, diathecus, especially our magnanimous NCERT, uses diathecus liberally, like I said. But do you know what is the real meaning? At the time of dehiscence, there is a strip of tissue between the two sacs. There is a division between the two sacs in both the lobes. When anther loses water, this division disappears and this becomes a single cavity filled with pollen grains. And to be very, very precise and correct, that is why a mature anther should be, I am saying should be, called as diathecus anther. Let's write it. Okay. So, just for the sake of precision, mature anther is diathecus due to a single cavity in both lobes. Okay, ma'am, got it. All good? Yes? Guys, I think you guys should also drink a lot of water. If you are really watching the class and writing down notes, because you don't have to dehydrate here. We don't want any dehiscence of concepts happening, do we? Okay guys, so how are you? How's life? How many of you understand the basics and liking the progression of the male generation that we are doing in this chapter? Is it making sense? Nikhil, I would inform the team. Yeah, crystal clear. You know, tomorrow there's one thing that skipped my mind. As promised to you, tomorrow at 8 p.m. I will have a special class for this chapter. In that class, we are going to do the most important, as well as we are going to deal with all the previous year questions of this chapter, so that you apply today's understanding and knowledge and really realize that you have done botany like never before. Does that make sense? Good idea? So 8 p.m. tentatively, right now it's 8 p.m. If there's a change in the time, and the link to that class is in the description box. Yeah, I want everybody to attend that class, but I'm going to add a little bit of more flavor to it. What we're going to do is, we are going to take your doubts as well. I want you to read at least NCERT, go through the PDF that you get at the end of the class, because you know, ma'am, textbook comes later, okay, and ask all your doubts. Whenever you study a chapter for the first time, it is mandatory, it is imperative to go through the chapter in the textbook and whatever notes that you get out of the class the very same day. Okay, immediately. So you can do that in the morning. Let's say the class goes on till late in the night, and then we take the doubts as well as the previous year questions. Good idea, serves well. Everybody is happy with that? Yes, you have chemistry class tomorrow. So we are going to do something about the timing. Even I realized it today. Yeah. You have physics class. I totally understand. Sir is going to take physics class. Okay. So we'll find a way out. Don't worry. Plus, I have kept my ecology class for Avengers batch at 5 p.m. tomorrow, wherein we are going to start with ecosystem. Just reminding you. Ruth, darling, I'll think about it. Good idea. Special class will happen on the app only. Cool. Thank you very much. Now we are going to move on to something called as microgametogenesis. Now, boys and girls, whether I am the primitive dicot who got pollinated, fell on the stigma, or I am the monocot who's carefree, who's smart and confident, and he's like, you know what, I'm going to develop further and at least reach my destination. What is the destination? Everybody write it in the chat box. It is to reach the male gamete. Male gamete is going to fertilize, pollen is not going to fertilize. Whatever happens, now we are talking serious business here, and that is microgametogenesis, the final development of pollen grain. And if you ask me, I call this as the male gametophyte. What is going to happen? This generative nucleus, focus on my words, I said nucleus, is going to undergo second mitosis. Because I'm N in nature, even if I want, I can't undergo meiosis. Please don't be confused when somebody asks you whether in this stage mitosis will happen or meiosis will happen. Well, think about the ploidy baba. You don't have to, you can't mug up every concept. If you try to do that, it's going to create a huge muddle. It'll be difficult to get out of that. Even if it takes time, please align concepts with common sense. I am N in nature. I can't undergo meiosis. So I go second mitosis. The result of mitosis is 2 daughter cells. I don't have to mug it up. The vegetative irregular nucleus is already present. He's patient. He's carrying a lot of food. What I get new in the picture, what happens is tiny, small, spherical, equally divided. There is no unequal division. Second mitotic division is an equal division. Don't assume. Okay, it's all scripted. So now finally I get 2 male gametes. Congratulations, we reached the destination. Was it so hard? Now boys and girls, if you ask me what my teacher taught me, this is still not a gametophyte, but it's okay. At your level, let's not make you wait further. This 3-celled pollen grain is actually your male gametophyte. Why? Why didn't I call the bicell pollen grain as male gametophyte? Outside in the world, people are calling pollen grain as male gametophyte, and here ma'am is waiting till the tricell, till the 3-cell stage. Why? Why? Think about it. Oh, come on people, think about it. Let's see who gives the answer. And now my task is, I have a very huge task now, because these male gametes, unlike bryophytes, unlike algae, in fact in gymnosperms they can be winged. These are purely immotile in nature. There is a reason, boys and girls, that angiosperms developed the act of pollination so much than any other group. If you talk about gymnosperms, there's only Anenophily. But these guys focused a lot, and we are going to see that the next topic, pollination, because they have to go to the female generation. Is that clear? Anybody who dared to or tried to answer why I'm calling only, I'm not agreeing to call anything else. Yeah? Any reasons? Because gametophyte is the generation which carries the gametes. Now we are going to talk about the structure of pollen grain. By that I mean the exterior, the walls. We are going to move our focus from the cytoplasmic contents and events, wherein you got the vacuoles. Now vacuoles are multiple vacuoles. Still there is a lot of food present. A pollen grain is rich in all those nutrients. Still you have the vegetative cell, you have the 2 male gametes, and the size of a typical pollen grain, as is said by NCERT, is 25 to 50 microns. It's usually a globular structure, globular in outline. But it is untrue. There are pollen grains of 100 microns also, 150 microns also. Mirabilis has 100 micron pollen grain. But if he asks as an MCQ, you would still follow NCERT and mark 25 to 50 microns. Now what we are going to do is, we are going to talk about this super scary sculptured layer called as the exine. Okay, so it is very spiny, sculptured. Let's look at the appearance before we go on to the composition. Spiny, sculptured, outermost. And then what happens? The next thing is the composition. This is made up of sporopollenin, which is a lipid rich substance. Okay, it is mostly a lipid rich substance. Okay, and it is the hardest or the most resistant organic material, like somebody said in the chat box. So whether you apply any physical agent, for example a lot of pressure or temperature, nothing is going to happen. It will not get destroyed. It is immortal, so to say. You apply a strong acid, alkaline, nothing doing. You want to give a fabulous, you discover a superpower enzyme, biological agent, nothing is happening. That leads, this point is related to the next point, which is that because of sporopollenin, we get microfossils of pollen grain, of exine to be very particular, of exine. And this again helps you to study various species. This study is called as palynology, wherein external structure of pollen grains is studied because of the immortality of exine. Is that clear? Now, this layer right here, okay, this is the exine, and we also have the, let's highlight it, we also have the inner layer which is intine. Very, very elastic. Do you see that? Do you see that? I'm going to use this inner continuous layer without any spiny appearance. This is your intine. When I say elastic, it is exactly made up of what collenchyma is made up of, the elastic tissue in plants. I spoke about it in anatomy. Yeah? So it is made up of pectocellulose. Pectocellulose. Why God gave it an elastic nature? Do you see the pores here? Because remember, the constituents that you got after so much of hard work till now, the 3-cell stage, the mighty male gametes, they have to reach the ovary after falling on the stigma, and that is done with the help of a pollen tube, which will come out through only through this gap, only through this gap, only through this gap. Going to talk about it. Okay, so this layer extends as intine, extends as pollen tube. Very, very important. And in all of this game, this guy is providing, the vegetative cell or the tube cell is providing nourishment. If your mind is thinking, what is he doing? Male gametes will. Now let's talk about the pores. God had to design pores because sporopollenin is unforgiving. It is not getting destroyed. Whatever you want, wherever you want, no. So these well-designed pores in the structure of pollen grain are called as germ pore. And the purpose of germ pore is just this, to absorb moisture and to let the pollen tube, poor thing, come out. Is that clear to everybody? Makes sense. Now germ pores, here we have shown 3 germ pores. They can be 1 also. It's tricolpate condition or unicolpate condition, not in your syllabus. 1, 2, 3. Maximum goes is 3. Is that clear to everybody? Done? Happy? Now there are some subtopics which are very easy to understand but give you very easy 4 marks. For example, pollen allergy. I just want you to focus on the keywords which will be asked as MCQs. You guys are ready for that? So first of all, it is the wind pollinated plants. Wind pollinated plants which cause this pollen allergy when they are trying to reach the stigma. They are not trying to harm you. And if wind is carrying it, it will go into the respiratory tract because they are so minuscule. They are only 25 to 50 microns. Good. What is the other name of wind pollination? It is Anenophily. Now, pollen allergy has varied symptoms in everybody. For somebody, it can just cause watery eyes, itchy throat. For example, I just get itchy throat. If I talk about my brother, oh my god, he gets watery eyes, he gets cough, he gets rhinitis, yeah, respiratory tract disorders. So acute symptoms is what I'm talking about. In some people it can cause chronic symptoms as well. For example, bronchitis. Now we are entering the zone of zoology. Acute respiratory disorders and chronic respiratory disorders, right? So the wide array of symptoms like rhinitis, or it can be simply an asthmatic attack. Yeah, people who have a weak respiratory system experience repeated attacks of asthma during this season. For example, grasses, which are Anenophilous, bronchitis, fever, anything. Now, when I don't have a particular set of symptoms, boys and girls, I come to call it as a syndrome, as is the case with AIDS. The symptoms of AIDS are not related to a particular organ or system. The patient can die of a kidney disorder, the patient can die of cardiac arrest, nephro, neuro, any system. So this is called as hay fever, which is not a disease. Please understand that in the core of your heart. It's not a disease. It's the culmination of various symptoms experienced during pollen allergy. And the third important MCQ of this topic is, who is the major contributor? Please don't mug it up, not allowed in my class. Who is the answers in the chat box? Since you guys are spamming, I'm trying to avoid the chat. Who is the major or the most invasive terrestrial weed, boys and girls? That is the major contributor. Who is that? Carrot grass, Congress grass, intentionally I'm not writing that. Thank you, Mayuk. You're not allowed to answer. Others, let others answer. Please. Guys, if there is a lag in life, in the session, kindly, faithfully, sincerely reload it. I'll give a break after the male generation finishes. Don't worry, even I need one. Guys, the question is not coming from carrot grass year after year. It is coming from the botanical name, which is Parthenium. Parthenium. It is also the most invasive terrestrial weed. There are other contributors, please don't forget them. This is a previous year question, I'm not giving any extra knowledge. There is sorghum. There is amaranthus. These are all weeds here. Chenopodium. Yeah, these are some of the common names that I've given you. I've not even troubled you with anything else. Is that good for us? Last topic of not today's class, before the break, we are going to finish this chapter of the male generation. It is pollen viability. What is viability? It is the ability to germinate. Definition based question has been asked. Ability to germinate after. What matters is, now my destination, I got the male gamete, now my destination is ovary, ovule, female gamete. After reaching the stigma, your NCERT mentions only 2 examples, that in wheat and rice, it stays only for 30 minutes. That is cereals, both the cereals. But in Rosaceae family, Leguminaceae family, and which is the third one? Come on, people. Fast. These are all families in your syllabus now. Isn't it? Other than Harshad, it's not survival. It's not survival per se. It's the vitality means being able to do the major job that it has to do. Okay. So, very, very important, Solanaceae family. Is that clear? This question is a previous year question. I am not making it up. Now, if you want to artificially enhance the viability, for example, people freeze gametes. There are egg banks, for example. Similarly, one of my favorite questions, ma'am, why is it your favorite? We have pollen banks, wherein you can keep pollen grains of rare species. Let's say there is a wheat variety, okay, and it's very special, it's very high yielding, it's disease resistant, it's beautiful, and the horticulturist is like, you know what, it's going to get over in a few years, people are over exploiting it. You can still preserve the pollen grains in pollen banks for years and years. You keep them in a very low temperature of minus 196 degrees Celsius, and this, my dear students, is called as cryopreservation. Are we good till now? Cryopreservation. Good? Happy? Happening? Yes? No problem at all? These are the questions. I am giving you only what matters. Only what matters. Other than that, now we will move on to the female generation. One thing I want to tell you is the pollen products. Okay, so pollination is usually done by bees, and when bees do pollination, we attain not only pollen grains but also bees wax. Not important, but this I told you why they are eaten by athletes and horses, for the sudden inflow of energy. Why? Because they have all the macronutrients. They have carbohydrates, unsaturated fatty acids. Yeah, carbohydrates around 50%, fats around 25%, approximately, there is a range, and proteins, the rest 15%. Is that clear? Makes sense to us? Cool. So, we will resume the female generation. This is just a repetition of your class 11th at approximately, I don't even have the, my phone is also off, at approximately 8 p.m. Does that work for us? Vijay, good thought. Good thoughts, good things. See you guys at 8 p.m. we are back. Everybody, please re-enter the class. Let's move on to the female generation, everybody. It's easier. Good news is it's easier than male generation. No, I'm not smiling because that's not the truth. But usually this happens. This is the experience of everybody. Once you have spent your time, since you got to know about the basics of what is sporophyte, what is the ploidy, when does meiosis happen, mitosis, what is the aim, female generation seems like easier and similar to the male generation. And I'm not kidding, it's easier. Now boys and girls, female generation, when we describe the floral characters, and I'm going to repeat this fashion in morphology as well, which is the next point of discussion, chapter of discussion, it is given more importance. The female generation is always given more importance. A female generation, whether it is animal kingdom or plant kingdom, this is the rule of nature, will never focus to increase in number, but it will focus on differentiating and developing its structure, bodily structure. Because always remember this, whether it's human reproduction or plant reproduction, yeah, because it has to not only receive the male gametes, but it also has to be the site for all further future events. Does that make sense? No? Yes? Sort of? Kind of? Because it is the female body where the fusion of gametes and the so-called, yes, in case of plants also, the pregnancy happens. And the next diploid generation also develops within the parent female body. That is why whenever we spoke about the gynoecium, the G, it was 1 or 2 or 3 carpels. As compared to other floral parts, androecium, sepals, it will be a pentamerous flower, it will be a trimerous flower, talk about liliaceae, talk about fabaceae, talk about, is that making sense? Don't know families, yeah? The Priya, beta, break is already over. Look here, use your beautiful eyes, it's already over, yeah? It was a short break, I realized it after it was over. Like, but if you guys will be nice, I'll give a longer break next time. Okay, so this is all class 11s. Please keep your pens and papers ready and please keep sharing this session with everybody, because you don't want to miss the technicalities and the precision of concepts presented to you in this session. Now, just like stamen was the microsporophyle or the leaf of gymnosperm that modified itself to take part in reproduction, similarly the carpel, or in other words gynoecium, which is the fourth whorl, G, like I said, represented by G. Pistil is not represented by P. That's why in the beginning of the session, at around I think 6:05 p.m., I said today you need to focus on the whorls. So we represent as G. Superior ovary, inferior ovary should ring a bell. Now when I said 1, I said 2, or it can be this. What am I doing, boys and girls? Come on, remove the dust. I am talking about a pistil, an ovary being monocarpellary, as is this case, or bicarpellary, as is this case, tricarpellary, as is this case. Am I very clear? Yeah. Similarly, when they are fused, we call it, what do we call it? Syncarpous ovary. This is all class 11th. Yes. And this is called as the carpels are, the pistils or the unit of gynoecium are free. They are free. Especially at the level of ovaries, they should be free. This is called as apocarpous ovary. Yeah, Michaelia and Pepe were the examples. Now, why do I keep saying ovary? Not just I, but the textbook or the botanists. Because ovary, like anther, boys and girls, here is the, you got it, it is the major site of action. Like I said, you can call stamen as microsporophyll. I allow you to call pistil as megasporophyll. Remember one thing, don't forget the basics. If any time, any term in this chapter or any chapter for that matter, plant kingdom, morphology, if something very basic spoken by any teacher on the planet is troubling you, is creating an itch, a nerve, go back to your basics. Don't leave it right there. It's going to grow like a cancer and take away all the marks that you could have scored otherwise. So, mega is always for female, like micro was for male, and sporophyll is generic, signifies the homology of leaves, taking part in reproduction, gymnosperms, all of that story. Now coming back to the point, this is like anther. It is the, akin to anther, it is the fertile part here. And when I say fertile part, that is the real sporophyll. I explained the entire sequence to you. You want to do it again? Let's do it again. So, now I am just changing, I am replacing micro with mega. Please keep looking at the screen. Microsporophyll. It has the responsibility to develop sac-like structure called as mega. Keep saying mega because we are in female generation. Megasporangium. And the fate of Megasporangium, poor guy, is just an empty sac, is to develop, don't forget the basics, the life cycle, Megaspore. Are we together till here? This is very easy. And Megaspore follows the tradition. It forms the Gametophyte. And the same thing, Gametophyte, what does that do? It has no choice to produce the haploidized gametes. What is the fate of gametes? They fuse together to form the diploid zygote and the life cycle repeats. So what do we study? This you do in class 11th. When you study everything about pistil. When you study, this is the diagram of class 11th, Baba. I have just shown it here to remind you that we studied the presence of locules or chambers. They are not structures, they are empty spaces. Remember unilocular, bilocular. For example, if you open up a pea, it's a unilocular ovary. If you open up, if you cut a lemon, it's a multilocular ovary. So many chambers are present. So, now here what you see, this one, this is nothing but a very cushiony tissue, a very nutritive and cushiony tissue. You hear about it in human beings also. This is nothing but the placenta. This is what? Placenta. Class 11th guys, class 11th. Placenta. If somebody is not mentioning it categorically that placenta is made up of da 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 da, you will assume where nobody goes, who goes? Parenchyma. Okay, so it is a parenchymatous tissue. What is the function? Nutrition. And ovules are aligned to this sporophytic generation, to this sporophyte, sporophilous structure. Yeah. So now ovule becomes the megasporangium. What I am going to take is a single section of ovule. Make sense? And we are going to study from outside to inside. Whenever you are studying a complex structure, be it zoology or botany, structure of heart, structure of kidney, lungs, always follow the sequence, inside to outside or outside to inside. Okay. Now let's ascertain the fact that ovule is precisely called as a megasporangium because a lot of things are going to happen here. But it is still a diploid generation. Okay. Now I am going to miss something deliberately here. I am not going to write it till you feel it, till you realize it. Yeah, eyes on the screen. So now this guy here we just saw was aligned to this very nice cushiony nutritive placenta. With me? The stalk, eyes on the screen, the stalk of this globe-like structure. Point number 1, globular structure. There is no star-shaped ovule. There is, please don't assume things, okay. So this is an essential. Boys and girls, eyes on the screen. This stalk which attaches the entire globe with the nutritive tissue, there has to be a connection, is called as the funicle. And there is no overestimating of it. That's it. It is attaching. There is attachment. There is a connection. Between whom and whom, that usually is asked in the statement based questions and goes for negative marking if you are not paying attention in life. So remember, between placenta and body of ovule. Now if I am making a connection, I need blood vessels, na baba? Oh come on. Like you study in embryo development, in human reproduction. Don't think I don't know because I don't take the topic. There is immense amount of vascularity which is in place. But you know, plants are intelligent as compared to humans and simple. So there is a vascular strand again running throughout the funicle. Don't miss that. Okay. So it is not only attachment but vascular strand providing nutrition. We are very good. Now, funicle on one side is attaching to the placenta. Yes, this funicle when attaches to this globe-like structure, I am not naming it right now, forms a point, not a structure, just a point called as hilum. There is a sequence in which I am telling you the structure. Please follow that when you revise, study or teach anybody. Whatever you want to do with knowledge, but do it rightfully, not half-baked, non-sequential. Hilum is not a structure. It is simply the point of attachment between whom and whom. Please make a note of that. And how do you make a note of that? Not by making a note of that. You actually realize it in the diagram. Always do that. So now funicle is point of attachment, sorry, hilum is point of attachment of funicle to this body of ovule. Yes, the weight is over. This body of ovule, actually precisely this body of ovule, which is a huge mass of cells. Do you see that? Parenchymatous again, nobody mentioned, nobody said anything, something else is happening. Assume parenchyma. This is called as nucellus. Okay, simply the body of ovule. We are still blank. What am I trying to say? This body of ovule, boys and girls, I just want you to appreciate what is happening. This is the body of the ovule. It is going to be, if it is parenchymatous, whenever in future, present, past, future, somebody mentions parenchymatous, primary function, storage of food. Storage, going to be food laden. So we got rid of all that layering in Anther, right? Epidermis is doing something, middle layer is doing something. Now it is lined by, choosing colors is the, it is lined by, just focus where the pen is going. Do you see that? And here they are not present, and the same goes this side. You see that, what's happening? No ma'am, we can't see anything. Okay. Yeah. So these layers, just like peels of onion, are called as integuments. If I have to be botanically very precise, very correct, which might not really affect your NEET MCQ, but your comfort and your assimilation power of the concept. That's too much, ma'am, we don't know what assimilation is. How you digest the concept. Yeah. For the ease of that, you call ovule as integumented Megasporangium, but you call nucellus as a Megasporangium. Okay. So what I wanted to add was precision of integuments. Is this clear? Makes sense? No problem? Okay. Now we are not done here. It's not that easy. Acha, tell me one thing, is it 2n structure or this is n structure? I think it's 2n. Is this 2n? Whatever we are studying. All of this. Thank you, Laharika. You are a darling. You always make me happy with your answers. This whole thing here, I think it's an n structure, right? No, it's not a 2n structure, is it? I don't know. So now we will continue with the structure. Okay. Now did you notice that integuments are actually absent only at this point, and rest of the globe, rest of the nucellus is covered by integuments. This area, boys and girls, where if I have to define structurally, integuments are absent. Integuments are absent, very important, and this is the only pore or only opening in an ovule, called as micropyle. Are we together in this as well? Spammers, you are not at the right spot. But anyway. And right opposite. See, as a human being, we have to do something in life. Spam nicely. So now, here what's happening? What do you see here? You don't see any layering. You see the layering starting from here, somewhere here. At this point, at the top, I don't see any demarcation of the layering because actually the integuments are starting from there. They are absent here. They are absent here, but they are arising from here. So structurally, it is called either the point of origin or we can say point of attachment. Yeah, origin of integuments called as the base of the ovule. Very important question. Or chalaza. Now I'll tell you one thing, entire female generation walks and talks and happens and around this polarity of this globular structure. Just like the North Pole and South Pole, you have a pole number 1 which is the chalazal pole and the opposite is the micropilar pole. Everything that you are going to study now will be according to this polarity. If you don't take care of polarity or forget the polarity, it will be difficult in life. Okay. This is also called as the apex. Micropyle is also called as the apex of the ovule, which is obvious. If this is the base of the ovule, then this is the apex of the ovule. But there's a very, there's something not right here. There's something not right here. I've been saying that this is the base and it rather looks like the apex. And I've been saying this is the apex and it's present at the base. Okay. Now listen to me. The ovule that you see in front of you, the ovule that you see in front of you is an inverted ovule. This is it's a little confusing in your textbook, so I'm mentioning it here. The orientation or the structure that you've just seen is of anatropous ovule. Anatropous or inverted ovule, which is the most common ovule present in angiosperms. There are certain other variants, and when ovule was first formed on this in this creation, that is when chalaza was present here as the base and micropyle was present here, just the opposite of this, that was the primitive ovule in some primitive families. So that is called as orthotropous ovule. So you can write it here somewhere. Straight ovule is orthotropous. Is orthotropous. Okay, but what do we study? We study anatropous. Is that clear? Now this pore is the only pore. This means that before the break, you form two male gametes which will be carried and one vegetative nucleus. Two male gametes, one vegetative cell, they have to be carried by the pollen tube. Pollen tube does not have permission to enter from anywhere else in the ovule but from here. This is the biggest importance. Okay, so we are going to add it here, very important point. Point of entry for pollen tube. If you remember, the sporogenous tissue, again very similar to the nucellar parenchymatous tissue. Sporogenous tissue was also food laden parenchymatous tissue. Now you see a structure here, this must be troubling you. I am deliberately not speaking about it. What is it? But you must have guessed it that this has got something to do with the next generation. And we can clearly see that this next generation, and now you guys are wise enough, or the gametophytic generation is oriented towards the micropylar pore, micropylar end. Okay, so this is nothing but the female gametophyte, which we are going to study just like male towards micropyle. Happy? At ease? No problem? Now, in sporogenous tissue, almost all the cells started acting like PMC or MMC. But here, only one cell, or maybe a little 2-3 cells, usually one cell will enlarge itself, have a dense cytoplasm, good prominent nucleus, and act like the megaspore mother cell. Megaspore mother cell. Okay, now that is why I gave you PMC, because here also the short form is MMC, right? What is the fate of MMC? To undergo megasporogenesis just like microsporogenesis. Yes. So, what is megasporogenesis? It is the formation of haploid megaspore from MMC by meiosis. Now, boys and girls, the added factor in female generation is at every step, everything will happen according to the polarity. So now, when this megaspore mother cell undergoes meiosis, there's no other fate of it. It is going to produce 4 megaspores, okay, or the tetrad of megaspore. In male generation, I have several types of tetrad, which is not in your syllabus now. It used to be. Yeah, tetrahedral, isobilateral. The common one is tetrahedral, followed by isobilateral. Linear is not seen. But in female, what you see is a linear tetrad. Now, eyes on the screen. If this is the micropylar end, like we just saw, I am not changing the polarity, and this is the chalazal end, like we just saw, what is going to happen is 1, 2, 3. Count 3 megaspores from the micropylar end. Count 3 megaspores from the micropylar end and tell them to go home. That is, they degenerate. Not mentioned in NCERT. You will mention it now. Kindly write it, sweetly write it, sincerely write it. And the one at the chalazal end, because what did I tell you in the beginning of this second session after the break? Female generation evolution has happened in a way that it is not focusing on the increased number. I don't need multiple female gametes. I want to make one egg. Yes, but in male generation we need as many gametes as we can to increase the chance of fertilization. So, this one here at chalazal end, you see the functional megaspore. Are we good? Got it? Just like microspore, there we got millions of microspores. This one will also be N because it is a product of meiosis. So there is no chance of undergoing a meiotic division again. Now whatever I have to do in life, I want to do it by mitosis. Yes? So now comes the next step. What is going to happen to the functional megaspore? Microspore developed into, microspore had nothing to do, we said he's not good for anything, he has to undergo development, there is development of male gametophyte, etc. So similarly, megaspore has to undergo the development of female gametophyte. And the good part is, there is no problem here. Female generation is very simple, sweet. There is no pre-pollination, post-pollination because she doesn't have to travel. Male generation had to travel. No confusion. Simply, mitosis will happen. And now, the good thing is, the mitosis is also free nuclear, not unequal. This is one of the favorite concepts that I have. Free nuclear divisions. What is a free nuclear division? When karyokinesis is not immediately followed by cytokinesis, that is wall formation. So what will you get? Let's see what you get. So if this is the functional megaspore with its one nucleus, I undergo a division, and trust me, here please don't forget the orientation, the polarity. You still have this and you still have this. The moment you forget polarity in female generation, questions are going above the head. So now what is going to happen? This is going to happen. This is the first division. Let's do the second mitotic division. Okay ma'am, we'll do it if you say so. This is going to happen ma'am, no problem. Okay. Female is like, you know what, my army or my team is not enough. I want more people to take care of me while I wait for the male gamete. So happens the third mitotic division. Everybody? And finally, what do you get? Are we writing? Yes. Finally, again, if you do the third mitotic division, have your eyes on the screen. If you do the third mitotic division here, you're going to get 4 cells each. 4 nuclei each. I'm not forming any walls around the nuclei. Okay, so NCERT doesn't even highlight it, but the examiner catches on it. These are free nuclear divisions to begin with. What do I get? I get a structure. Within the ovule, something is happening. Chalazial end has 4 nuclei, microbial end has 4 nuclei, and now everybody is happy. Floating in the cytoplasm. Yeah? There we go. But then the plant says, I'll draw it for you and then we'll go to the NCERT diagram. And now the ovary, who's the boss? Who's the parent? Tells ovule, hey, what people are doing? They're doing so much of notorious things, so much of ruckus. How can they not have walls around them? How can you allow them to just lie freely? Not perform cytokinesis? So now the ovule tells this structure, which is going to be the female gametophyte, that you know what, you have to do something about the organization of nuclei which you have formed. Very well done, you have undergone 3 mitotic divisions. What I am telling you is the most commonly asked question. How many mitotic divisions will take place? How many mitotic divisions will take place? How many divisions will take place in MMC? Then you will have to add the Meiosis also that happens in the beginning. Don't skip the steps. It will cost you negative marking. So now what happens during, what I am going to tell you is the organization of embryo sac, which is nothing but the female gametophyte. This is not embryo sac. This is a ill-developed. It is getting scolding. Now here, when ovule tells them to behave properly, this guy here, do you see that? Eyes on the screen here. Eyes on the diagram. This guy here at the chalazial end, and let's say this guy here at the micropilar end. Okay, let's take the same. Then you guys will assume, oh, it's the second nucleus only, ma'am. That is the fourth nucleus. No, 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 no. Please don't assume things in science. This guy here, they are the most notorious. They are not listening to anybody. All of these guys are ready to align, okay? They are ready to organize. But these two are troubling everybody. They are not letting anybody do the right thing. So now ovule says, get out. Get out of the class, spammers. And this guy migrates in the center. And this guy migrates in the center. Are we together? And they come in the center. They don't have a choice. They were disturbing everybody. But since they have come from two poles of the structure, pole, they come to be known as the polar nuclei. It is as simple as that. I have no clue why people mug this up so much. And now the three which are remaining, let's tell this guy to go. He got punished. This one got punished. Somehow I also got punished. I have to draw it again. So now how many are remaining? 3 are remaining. They will start forming walls around themselves. They will start forming walls around themselves. They were always obedient. See, this is not how it is. But I am just telling you the basics. This. And now I have 3 nuclei at the chalazial end, 3 nuclei at the micropilar end. And when these 2 polar nuclei get in the center of the going to be embryo sac, they are like, you know what? Together we can trouble large masses of people. We can do real great things. Let's unite together. And then what happens? Let's go to the NCERT diagram, which I have not labeled for a reason. You're able to appreciate? We got this. 1, 2, 3, 4. 1, 2, 3, 4. C and M. Happy? Ordered to be organized, because you can't go on like this. Organization happens. This is not given like this in the textbook. So it would really benefit if you listen. Yeah. So at the micropilar end, the 3 group of cells, and remember, everything is N. Everything is N at the micropilar end. They are called as egg apparatus. They are called as the egg apparatus. Yeah? And these 3 are called as the antipodals. And these 2, do you see there is a fusion that has happened here? They decide to come together and fuse together. Okay? So it becomes a single nucleus. I didn't say cell. There's no wall. This single nucleus formed by the fusion of two polar nuclei is called as the secondary nucleus. I promised that gametophyte generation is going to be completely haploid in nature. But this is a paradox here. Female generation has one nucleus which is diploid in nature. It intended it to be haploid, but these guys just didn't listen. Okay. Now, as the female gametophyte matures further and gets ready to receive the male, it develops a wall around itself. There we go. And now this 2N or diploid structure, which is larger than other cells of the skein, although it doesn't look like that, but that is the case, it is called central cell. This is exactly the diagram of NCERT. Central cell. Okay, and this is the largest cell of embryo sac. Where is it present? Not on any poles, it is present in the center. On these poles, all three are antipodals. Antipodal number 1, 2 and 3. But here there is a catch in the story. There is a catch in this story. Have a look at this egg apparatus. It is not like this, okay. It is arranged somewhere like this is your egg, and this is synergid number 1, and this is synergid number 2. Yes, have a careful look. It gives you the idea of a triangular outline. This is how the egg is protected by the synergid. Okay. Now, there is one more thing which is a little outside the syllabus theory wise, but NCERT has shown it diagrammatically. So, I don't want a rainy day where examiner says, hey, I asked the question because it's given as such in the diagram. And you guys are like, well, this was out of syllabus, most difficult paper. Oh my God, what happened? Why? How? When? If. What NCERT has done, and it really excites me. Okay. This is the microbial end. We are maintaining the polarity. Have a look at the screen. At the microbial end, you don't see the nucleus of the egg. The nucleus of the egg is directed towards the chalazal end. But if I look at, and this is NCERT talking, but if you look at the synergids, don't mind this, why did you draw like this? This is a hook. Every synergid has a hook, don't worry about it. The nucleus of synergid is central, it is central, or it is rather present towards the microbial end. So, this is a opposite orientation. Do you know why? If anything happens to the nucleus of egg, any damage happens to the nucleus of egg, then the only chance of fertilization is gone because queen is one. So, these are like the bodyguards. These are like the bodyguards. This one is fine. Guys, if you think this is too difficult for you to understand, it is okay. But I am telling you, one day, don't let this happen to you. This is given in NCERT. This outline which is triangular, it is called as piriform. Remember, I want to connect this concept with another concept of plant kingdom. Piriform gametes found in brown algae. The same thing. Yeah. So, these are triangular outline of egg apparatus, wherein you have the queen or the egg. I am informally saying the queen, botanically you don't call it the queen, and these two, okay, are the synergids. Whatever force or damage, because this is the micropilar end, guys, pollen tube is going to come with a lot of speed. It might collide. It will never be allowed to collide with the egg directly. Never ever. Because that force is going to break the nucleus of the egg. So, God has kept the nucleus of the egg away. And invariably, the pollen tube first has to deal with the bodyguards. And their nuclei have been kept here. Head-on collision. We'll speak about that. Okay? One more thing, these finger-like projections that you see, these are not structures. The tissue has modified itself. This wall has modified itself. But filiform apparatus, finger-like filiform, these are often used in zoology also. Filiform. Filiform apparatus will never have anything to do. See, nobody is allowed to talk to the egg. Nobody is allowed to talk to the egg. So, there is a true and false MCQ that was asked in previous year, and we are going to do previous year questions in the special class tomorrow. The link is given in the description box, and I will show you how examiner has twisted the question by just writing that filiform apparatus. I am going to tell you the exact statement. The question is right in front of my eyes in this very moment. Filiform apparatus directs the pollen tube entry towards the egg, and masses had marked it as a true statement. No, filiform apparatus doesn't even know the egg. Filiform apparatus are these finger-like projections. They are present at the foot of the synergids like this. And it tells the pollen tube, you know what, you can enter either here or here. Talk to this bodyguard or talk to this bodyguard, because there's no chance with the queen. That's how it is. So, I'm going to write it here, mention it. Direct pollen tube towards, not the egg here, towards synergids. Are we good? Everybody, did you get it till here? Very good. You guys are very creative, yeah. Are you guys okay? We can move on to the next topic. Is this organization? Now, you might ask me, what is the function of these antipodal cells? Okay, so I'm going to write that they are the vegetative cells. Does that remind you of anything? There was a vegetative cell in the male generation also, no Baba? The term vegetative has been used to signify that, it is really not involved with the gamete, with the act of fertilization. Okay, so similarly, this vegetative cell present at the chalazial end is not going to take part in what is going to happen now. Yeah, so you got your destination. Wow, you have the gametes of both the generation. And here comes a previous year question of 2022. Can you tell me the answer in the chat box, people? Let's read the question. In general, the egg apparatus, very easy question. Just trying to make you understand that examiner is not against you. If your concepts are clear, easy questions are asked in the paper. If your basics are not clear with such huge chapters, then you might face a problem. In general, the egg apparatus, I just spoke about egg apparatus at length, of embryo sac and angiosperms consists of, answers in the chat box. Mayuk, you are not allowed to answer, Baba. When you have studied the chapter 18,000 times, you're not allowed to answer. Let others also. दूसरों का भी सोचो यार. Everybody is stuck with the A answer. 1 egg cell, queen will be 1, 2 synergids, bodyguards. Don't miss out on 3 antipodal cells and 2 polar nuclei. They were pushed outside, but they are doing good things in life, bigger things in life. They didn't just sit there. They fused together to become the largest cell of the embryo sac. Isn't it amazing? Yeah, don't forget, these are all potential MCQs. So now, when I have both the gametes and both are ready, both are sexually receptive, there comes the act of pollination, which is actually required for the male generation particularly. Yeah. So, do I need to tell you the keyword? It is the transfer of pollen grain, and guys, it can be 2 or 3 cell. Please don't assume. Ma'am, will it be 2 cell? We already spoke about it. Transfer of pollen grain from the male parent to stigma. Now, eyes on the screen, everybody. Amazing, yeah. People, again, what do we do? Now, I want to say something which is very important, but I am not going to just serve it on the platter. Ready? Now, when I say this transfer happens, it can happen in 3 different scenarios. Yeah. Common sense, please. One scenario is that it is very simple and it happens in the same flower. Focus on the keywords. I am not giving you page long definitions or concepts. I am trying to make it easy for you. Same flower. Another scenario is I don't want to reproduce within the same flower, but I choose another flower and the plant is same. So the keyword is same plant. Happy, healthy, no problem. And now, you know what I say? No, I want to cross the boundaries. I don't want to reproduce, fertilize the same within the same plant. Let's create more variation, and I go to a different plant. Okay, so the key word here is different plant. To help you focus on what's happening really, genetics is changing here. In this kind of coming together or marriage, genetics is changing. Okay? You know, you have studied genetics at length. You know what is the consequence of this. You know what is the consequence of this. Within the same flower, absolutely nothing to do with any variations, any genetic variations. Now, I am not heading into that domain at all. Let's give it terms. This is called as self-pollination or autogamy. Gamy is marriage, gamos is marriage, self is same, auto is self, yeah, auto is self. Now here what's happening We'll come to that. Let's go to the next extreme. In zoology, you must have studied, I don't know, I don't remember, we used to study about grafts, xenografts for example. So when I am choosing another plant, this is going to be called as xenogamy. Okay, xenogamy, or in simple terms, cross pollination. I am picking up the genes of another plant. Is that clear? Now what is this? This is a negotiation between the two phenomena, and you will see how. No genetic variation will take place because the plant is same. More often than not, people write this scenario as a different flower, 99% of the people. But what I am trying to do here is, when I write same plant, this keyword reinforces another fact in your beautiful heads. It's good to assume good things, isn't it? So this becomes genetics. Let's use the same genetically same, and this comes to be known as Gametogamy. Gametogamy. Are we together? Yeah? Gametogamy. But there's one of the lines given in NCERT very close to my heart. Ecologically or functionally, this is cross-pollination. See, I'm compiling all the theory and giving you here the entire mind map, like you guys like to call it. Huh? So, what do we write? We write it ecologically or functionally cross-pollination. Ma'am, if you are really into this game of NEET, I'm not going to lie about it. No. Your mind should be asking this question, ma'am, what is ecologically or functionally cross-pollination? Why? The moment your brain stops asking the why of things, understand there's something hugely wrong. Okay? So, understand one thing, that even if the flower is present on the same plant, the problem is I need to transfer the pollen grains from one flower to another flower. So, functionally, that is, it has to be brought about just like cross-pollination. The distance will be less. So, let's say this marriage is happening between two countries, people of two countries. Okay? Here they might have to travel between different states in the same city, in the same country. But the traveling happens. So, genetically, this question, oh my goodness, so many times, genetically same, ecologically. Ecology means how am I dependent on other organisms? So, ecologically it is dependent on agents of pollination. Now, one more thing again, if you guys want to know, I have often seen people using the term allogamy and xenogamy as synonyms, which is not precise botany wise. Okay? So, again, for the sake of precision, you can ignore it if you, this hasn't come as a question since a long time. There is something called as allogamy, which is not synonymous to xenogamy. Allogamy is simply Gametogamy plus Xenogamy. If a plant is engaging in Gametogamy and cross pollination, which most of the plants would do, that is why I tickled your brains a while ago. Every plant will choose to undergo. Now you know the terms very well, Xenogamy, because I have to evolve. Is that clear? Now, what is the question that is asked out of all this? What I just told you is the tip of the iceberg. NMC has deleted certain important topics which broke every botany teacher's heart. Transport is done, mineral nutrition is done, secondary growth, yeah? But it did not ever touch pollination since I was a student. Well, we used to have at least 1 or 2 classes on pollination. I have, in fact, 50 pages right in front of me. But we are going to do it in a concise manner, and I'm going to give you all the questions that can be asked. When I talk about autogamy, ladies and gentlemen, what you have to focus, contrivance. Ma'am, we don't know what contrivance is, ma'am. Come on, yeah. It's very much in your syllabus. Are the devices or the conditions for something to happen? Okay, you have a topic in NCERT called as outbreeding devices. Yes or no? Outbreeding devices are contrivances or devices or facilitation of cross-pollination. Similarly, NCERT does not shy away from discussing the detail of this topic. I'm warning you, pollination is something you guys don't think I don't know. I'm on this side of the camera, who knows? I know what you skip, what you like to skip. You breeze through it. You just think, no, pollination is not my cup of tea. But these are the potential questions. And we will use common sense. Yes, you guys know it. The same inbreeding depression, the same concept of genetics will apply here. So no plant, I can tell you, will undergo pure autogamy all its life. It will cease to exist. But sometimes autogamy is done to create pure lines and save certain superior characters. Again, we are going into the boundaries of genetics. So the first thing I need for autogamy is bisexuality. Yes, or a hermaphrodite flower, which, good news is, most of the angiosperms are. So the first thing is bisexuality. Second thing I need is homogamy. Very important question. This term is not given in NCERT, but the phenomenon is given. Even if a flower is bisexual, it has the required organs, male and female, carpel and stamen. Very nice, healthy. And they are producing the gametes also. No problem. But if their sexual maturity is not synchronized, or the receptivity is not synchronized, autogamy cannot happen. Synchronization of sexual maturity, which simply means that pollen grains should be ready to release and stigma should be receptive. Then comes the fact that if I don't let the organs be exerted or open, autogamy is a short mechanism. That is, I am not even letting the flower blossom. There is a difference between bud and a flower that does not open. It is simply that the petals, the corolla or the sepals, they are not allowing the stamen and carpel to be exposed. Closed flower. Okay. So, this is cleistogamy. Again, NCERT speaks about it quite nicely. Cleistose is closed and gamy is coming together, marriage, fusion. So, this is closed flower. Now, boys and girls, no angiosperm will have only cleistogamous flowers. Together with me. So, mostly what you will find is present along with chasmogamous flowers. Chasmogamous flower is just the opposite of cleisto. Chasmo is open. So, they will mix and match. Later, sometimes what happens is the breeding season is going on, cross pollination has happened or Xenogamy has happened because the flower was open or chasmogamous, and then the flower is like, you know what, I am getting bored, let's do something different. Flowers close, cleistogamy. Not kidding, this actually happens in, I am going to give you the examples, very, very, very important. Previous year question. Example, Commelina, Oxalis and Viola, common pansy. Makes sense? No problem? Happy? Giving you only the essential information here. Otherwise, pollination is a huge game of concept. Okay? Does that make sense to us? And lastly, this is rare. The last condition is bud pollination. Bud pollination. It's a floral bud. Okay, it's a floral. Usually seen in, it can be seen in wheat, tobacco, potato. Very rarely. Okay, so tobacco is bad for health. Potato is very good for health. Both belong to the same family. And certain varieties of wheat. And this is all evolution, guys. Now we will talk about xenogamy. Let me be very honest with you. If I am the examiner and I have to choose one MCQ from contrivances or devices, if you want to make it simple, I would choose this. This topic is particularly pinpointedly mentioned. What am I talking about? Outbreeding devices. Please make a note of it before you regret later. Please, if you haven't been paying attention, give me your 5 minutes. This is a short question. And I feel it's going to be asked. Very well done. The chat box looks so neat centric. Bipin Chaudhary, you would be delighted to know that I missed your message earlier. I just missed your message. Somehow I have this superpower of filtering through the good messages, but yeah. Thank you for your concern. You don't have to worry. Please study. Just focus on the quality education that is given to you. You have reached the right place, meeting the right people. Now what matters is what you do. Okay. Now going further, before I miss out on all the important concepts, I am going to go a notch outside NCERT because the topic deserves it. Let's do that. Okay. Now common sense people, yes, the advantages and disadvantages will be same like you study in genetics. Xenogamy will be genetically variable, causing evolution, new characters, wow, amazing. But disadvantages, chance factor is there, right? Fertilization might not happen, a short seed set is not there, a lot of wastage, it's very expensive. The plant has to work really hard for making it happen. Certain recessive characters or harmful characters may pile up. I know you guys can do that because you guys love genetics. So if I apply my common sense, and I spoke about bisexuality in the previous case, I can easily say unisexuality is a must. Or in other terms, it is called declining. If you want to miss it, you can. It is just a habit. Nobody will ask you. It's just habitual that we used to teach this earlier before NEET happened. So now children, there is a concept, there is a twist in the story which you guys often make a mistake at. Unisexual flower can have two cases. Let's make it clear once for all, because I don't see anybody making it clear, and this is very important for the health of botany. If unisexual flower, that is male and female flower are present on the same plant, and there is another scenario where the male and female flowers are apart, they are on different plants. This is a question that is asked repeatedly here. The morphology chapter of class 11th makes it very clear, but still we fail. This condition for a plant, I see everybody saying, including people who are delivering knowledge, unisexual plant and monoecious flower. Please understand the word sexual. Unisexual and bisexual terms are used for a flower, never a plant. The term monoecious, is used for a plant. Plant is not sexual. Okay? So now when this happens, it can only, the conceptual question asked by the examiner, this situation will only prevent autogamy but not gametogamy. Because if you remember, that is why I gave you the keyword same plant under gametogamy. Since you guys have so much to remember, life is so tough, let's remind ourselves. Same plant, gametogamy, genetically similar. Different plant, xenogamy. Let's go further. Now you can mention it with a lot of ease and digestion at play. Prevents only autogamy. And it does not prevent gametogamy. Like, why am I smiling? Do you want me to write even that? let your head also do some work. The only word states it all. But if you want to write it, please feel free to add that into your notes, because sometimes things happen. Okay, different plant. This is called as dioecious. Something is missing. Okay. Dioecious means when male and female flowers are present on different plant. Okay, let me give you the examples also. The example of this is from the chapter of morphology. Anybody who would be a darling and give me the answers. Example, very, very important. Cucurbits, for example. Coconut, for example. Here, papaya is the example. So, in this case, both Gametogamy, there is no chance of Gametogamy because the opposite gender is not present on the same plant. I will have to travel to a different plant, right? So, both Gametogamy and Xenogamy are prevented. Good, no problem at all. Just for your information and for better closing of the concept, papaya and date palm. Whatever examples you remember from morphology. Is that clear to everybody? This is the first concept of unisexuality, and now I am going to take another concept right here. I like to save the space. Now, the problem is, what if the plant is bisexual? Because mostly angiosperms are bisexual here. Then the plant thought during its course of evolution that I can follow the other thing. I can break the synchronicity between sexual maturity. So, you know what? If you learn the devices for autogamy, this thing is solved. So, homogamy becomes dichogamy. Easy. Why does it become difficult? This topic becomes difficult because mugging up, right? So, let's not do that. Dichogamy. Di is 2, okay? It is exact opposite. There is no synchronicity between sexual maturity. Let's write it in short. No synchronicity between sexual maturity. Even if the pollen grain is ready, stigma is not receptive. Now what happened? It's a bisexual flower and both are receptive. Both are sexually receptive at the same time. Then the plant is like, the plant doesn't want this union to happen. So it will try everything possible. Now you think like a plant. What to do here? They are ready. Make them hate each other. Okay? So the third one is self-incompatibility. Self-incompatibility, the question often asked is, it is more definitive than the other two. You know why? Because this is happening at the level of genes. This is genetic in nature. So genetically the pollen grain will not, even if it falls on the stigma, they are sexually mature at the same time, the stigma will not accept. Or in other terms, even if stigma is accepting, both sides can happen, S1, S3 and S1 or S3. You don't have to get into the phenomenon at this point, but what I am trying to say is, even if the stigma is ready, the pollen grain might not germinate. Good. Now what happened? Still in under the water. They are compatible. They like each other like nobody's business. What do? We set them apart. We don't let them meet at all till they forget about each other. And that is done by the fourth technique, which is heterostyly. Heterostyly. Ma'am, what is it now? Baba, I don't have to tell you, break the words. Hetero is different. Styly is stigma, style, ovary. But what it really means is, there is so much of discrepancy in the length of male and female, that male is not able to reach the stigma, not see stigma, or stigma is so short, male is somewhere else. Okay, so we'll write difference in length. Of whom and whom? Common sense? Anther and stigma. Good till here? Yes? Now there is one more, but I think I have told you enough. Oh, we have this space. We can do one more. There is something called as Hercogamy. Very, there is no chance that this will be asked. You know what Hercogamy is? That is the last resort plant has taken. One of these will work. The plant is like, okay, they are of equal length, they are compatible, they are sexually mature. Now what do I do? Interestingly, it will cause barriers, mechanical barriers, that the union will not happen. To the extent that sometimes there is a flap of tissue on the style. Pollen grain has fallen down. Ovary is like, oh, pollen grain has come, please let him come. Oh, pollen grain, you've reached finally. No, there is a mechanical barrier. It's seen in Calotropis, for example. Entomophily is full of all kinds of evolution that has happened in with respect to pollination, and that part of science of botany, what is happening in pollination, is still getting discovered. Authors are still writing books. Still there are phenomena which we have not understood, to what extent plant goes for its survival and to increase its reproductive capacity along with maintaining genetic variation. Plants are very pragmatic. Much more, okay, much more than human beings. Yes, I am biased because I love botany. Okay, so guys, now what we are going to do is we are going to focus on xenogamy, because this is the problem area. Why it is the problem area? I need a lot of expense to bring about xenogamy. That is, I need agents of pollination. Oh yeah, it's there. Agents of pollination. These agents, in the beginning when the plant started the idea of pollination, I was going to draw this only, but you can see there's nothing, there's no difficult concept here. So initially when plants decided, they said, yeah, we'll send it in the air, big deal. And then some of the plants were like, no, you know what, we'll do it in the water. Understand that we are talking about angiosperms. Why do I say that? Why do I say that? Because when we talk about plants, in lower plants, if I talk about algae, bryo, teredo, only choice. When I evolved into a gymnosperm, only choice. Generally speaking, I know a lot of gymnosperms who use entomophily also. Yeah? And I said lower plants. I did not say aquatic plants. Again, a previous year question, 2022, 2020. Hydrophytes or aquatic plants mostly go through entomophily. Every plant on this planet wants to go through entomophily because it's very, very good for the plant. Okay, we'll see how and why. So this is lower plants. Now, understand that the plant quickly gave up on this method of pollination. Why do you think so? Common sense. Yeah? Very wasteful. Whatever is in the air flowing through the wind, I have no control over it. Extreme wastage. Hydrophily. Extreme wastage again. Most of the time pollen grains are drowning under the water, or they are killed, they are gone, crushed with the water currents. Now someone in this batch of students should ask, ma'am, how can pollen grain be harmed or crushed? It has the super special sporopollen. Yeah, you should ask these questions from your teachers. Now these are the biotic agents. The plant thought, I need something reliable, otherwise I will cease to exist here. So when it went to the living or the biological world, boom, the group of animal kingdom, which is like in humongous amount covering the planet, it is the arthropods. There we go. Okay, there we go. 80%, if I am correct about the data. Okay, 80% of the angiosperms, and it's super economical for them. And then everybody else was like, why not us? So comes the next common, which is bird pollination. Ornithos is bird, they say. And actually every living agent, you and I, while taking a walk in the park, could be facilitating pollination. There are such microscopic structures. So this is actually zoophily. There is malicophily, for example. Chiropterophily is bat pollination. You know, bats at night fly for kilometers and kilometers and cause long distance pollination. So these are dispersive mechanisms. And this marks one of the reasons of success of these plants. Dispersive mechanisms. Now when I talk about Anenophily, it is the thing seen in grasses. All the cereals that are present in the kitchen, these are all, Poaceae is the largest family of monocots. So Anenophily will be seen in grasses, palms, advanced family as well. What do you think are the features of flowers? This is the question which examiner is interested in. So what we are going to do, we are going to take help from what we have been blessed with. That is common sense. If I want wind to transfer something and I want something to receive that transferred product, what should be the floral characters? Think about it. Apart from the fact that this phily, Anemo is wind, phily is love. So either we are talking about gamy or phily. Yeah? Largest amount, huge amounts, largest amount of pollen grain. In maize, for example. Yeah? Like I said, the family of grasses. The most wasteful process. Now this is, I'm talking about the phenomena. I want us to think about, rather than taking points from somewhere, the features. Now if I am talking about the male flower, First of all, my stamen should be exposed and overexerted, overexposed towards the wind. So you will come across two terms in class 11th, exerted and inserted. Not in your syllabus anymore. It's very important. Not everything that is out of syllabus is meant to be thrown away. Mark my words. Exerted means it is exposed above the foliage, above the level of sepals and petals. You can easily see them, and they're versatile enough. Versatility is another. Versatile enough to move along with the wind current. Yeah, let's write that. So, exerted stamen. No problem. Now, if I will release the pollen grains, can I make them very heavy? No. Wind carries dust with it easily. So, there you get the word, dusty pollen grain. Exerted stamen, then versatile stamen. What is a versatile stamen? That moves along with the wind. If I don't move along with the wind, wind is not going to carry the pollen grains. So, powdery, or rather add more to it, unwettable. Add more to it, non-sticky. Add more to it, light pollen grains. There we go. So much so, there is a plant called Utica. Is it Utica or Utrica? It's Utica, which shows gunpowder mechanism. Gunpowder mechanism. What is it? The pollen grains burst and release a grey colored or black colored mass or powder of pollen grains. Suddenly they'll burst. Yeah, like how a gunpowder, how a gun shoots. Okay. And then this is about the male flower. Right. Now, if I want to attract the pollinator, I need to give certain rewards to the pollinator. I need to look attractive. I need to have fragrance. These are the various kinds of rewards or attraction. These are not rewards. These are the ways that pollinating agents are attracted. Rewards are separate. I'll talk at length during biotic pollination. But in this case, abiotic pollination, you are talking to the inanimate things. So, needless to say, but this is given in your NCERT, they are colorless, odorless, nectarless. I don't think anybody would mark true to that they contain all this. Nectarless, small, inconspicuous. Hardly they can't be seen. Think about grasses. Just think about, that's why I gave you the example. Inconspicuous, packed into inflorescence. Why am I packed into inflorescence? Why am I packed into inflorescence? So that I release the entire amount of pollen grains at once and improve the chances of reaching the destination. Anther. Is that clear? So this is all about male. Now the one, let's continue. Okay? Any more continued? Let's ask for some severance. Any more continued? Female flower. Now, if you want to catch the dust, Every morning the maid comes, or maybe you are cleaning the room on your own. How do I catch the dust? With the broom. Done, dusted. Hair-like, feathery, fine stigma, and classic example is tassels of maize. You know, the corn, the cob that you get, those tassels or hair-like structures that you peel off because you're eating the seeds. So what you have is the female flower, not flower, it's an inflorescence rather, called as spathe inflorescence, matter of old times. Okay, so trying to tell you that the hair or the stigma, the styles are so elongated so that they can trap the powdery or the dusty pollen grains coming to them, and these are usually long distance travels. To catch pollen grains. Good. Female flowers are also exerted because they have to catch, they have to receive. They are uniovuled pistil. What do you mean by uniovuled pistil? There is only one ovule. Again, because I want to increase the chance of fertilization. Remember the aim of the process. That's about it. That's about it. So you know the feature of the pollen grain, you know the feature of the female flower, you know the feature of the male flower. Now comes hydrophily. This is happening in the waters. What do you write about it? It is the rarest pollination. Rarest pollination. Seen in only 30 genera of monocots. Rest everybody said we are not doing it. Okay, and it happens in two forms. Epihydrophily and hypohydrophily. Yeah, so what do you mean? What do you understand by epi and hypo? These are the common prefixes. Epi is above the surface of water and this is below the surface of water. Yes, everybody? Good morning. How do you do? Epihydrophily, hypohydrophily, that is below the surface of water. Now, if I am above the surface of water, common sense please, because the characteristic of the pollen grain will change. Okay. So, if I am above the surface of water, I need to keep the pollen grain floating. I don't want the pollen grain to drown. Hence, I am spherical essentially. And mostly hydrophily. Remember, I tickled your brain a while ago. I told you that why do we say that water currents will crush the pollen grain? Pollen grains lack exine. There you go. Why? To make it easy for them to float in the water and not drown. Xine is very, very heavy. Whereas if I talk about hypohydrophily, under the water, I need to fight with the water currents. So my shape is going to be ribbon-like. And I am going to be covered by mucilage, so that I become even lighter. Example, Vallisneria, Zostera. What is the common name of Vallisneria and Zostera? Anybody? Because question has come from that common name, and the diagram, the picture is given in NCERT. It is sea grasses. Why do you call them sea grasses? Monocots, hydrophily, aquatic, seagrass. Okay, and give me example of epihydrophily. If I am not wrong. Is that clear? Good. Now we are going to move on to entomophily. Yeah, we are going to move on to entomophily, which is the most common method of pollination. What do you think will be the characteristics of these flowers? There is one thing to attract the flower, the pollinating agent. Other thing is to give it reward when it comes. So this relationship is very, very mutualistic. Or in some cases it becomes obligatory as well. I spoke about this in my plus class yesterday, wherein I made you write everything about mutualism. Yes, the Avengers batch told you how it becomes necessary. Now, what you are going to write most common? In the course of evolution, I, the flower, became very pretty, brightly colored. I became large and showy petals. If petals are not showy, then the plant will make sure that sepals will become showy, bract will become showy. There are examples of that. But the plant will have some side of attraction. Okay, brightly colored, large, abundant nectar. This is the main, but we are not talking about the reward. Let's talk about this. Brightly colored, there will be a smell. I didn't say fragrance. I didn't say fragrance. Think about it. There's a smell and there is nectar, color, smell. Yeah. Now, this smell could be pleasant odor or foul odor. If I'm a jasmine flower, wow, nice. But if I'm a Rafflesia flower, then I'm going to like very, very bad smell. Then I'm going to emit very, very bad smell. Okay. So foul, this is a question that is asked. Rafflesia, hydrogen peroxide smell. Amorphophallus. What about Amorphophallus? Isn't it? Now the reward system, which is actually confirming the mutualistic relationship. The rewards are many. Either it will be nectar. Mostly every insect will visit the flower for nectar. Or it will be edible pollen grains. It could be edible seeds that I have to offer. Or something, oviposition. Now, boys and girls, whenever the relationship involves oviposition, I spoke about this in my plus class yesterday. Whenever the relationship includes oviposition, ma'am, we don't know what is oviposition, ma'am. Ovi's egg position is space. The insect is using plant space to keep its eggs safe, to incubate the eggs. In this case, the relationship often becomes obligatory because the life cycle gets entangled. The larvae, the young insects will not come out if the flower doesn't take care of them in terms of nutrition also. It's not only space, Baba. The flower will have to feed literally with the seeds also. This is seen in the fig species, like I said yesterday. Yucca and moths, Pronuba moths, obligatory relationship. It's fig and wasp. But on the other hand, we have sexual deceit also, isn't it? What is the example of sexual deceit? Now, there are almost, the most common insects are bees, butterflies, moths, wasps, beetles, yes? But the common ones, most common, pollinator is bee, 80% of the angiosperms, followed by a butterfly. Yeah, you see that in nature. My question to you guys is, why a bee is never seen around a red colored flower? As simple as that. Okay? Other than that, the flower has landing platforms, markings on petals to guide towards nectar glands. Children, let me tell you one thing very clearly. The nectriferous glands in an entomophilous flower is always located in a position that when the insect enters, it definitely disturbs or touches the pollen, the anther part. And when it disturbs that, the pollen grains automatically stick to the back of the insect. And the insect is like, wow, I just had the time of my life. I had very sweet nectar. It's a sugary nectar. Not realizing that there are pollen grains stuck to the back of the insect. So that brings us to another factor, na Baba? Which is, let's write it here. Let's use the space. Yeah, I could have been a little good with the space. Sticky, very, very important, huh? Sticky pollen grain. Now, in certain entomophilous flowers such as Calotropis, there is a pollen kit in which, you know, the insect carries the entire pollen kit on its back. And there comes the function of tapetum, which I spoke about in the beginning of the session today. Sticky pollen grains packed into a pollen kit. Children, tapetum is giving lipid to form sporopollenin and hence exine of pollen grain. We agree? We agree. Here also, what is the stickiness caused by? Pollen kit is made up of lipids and carotenoids. Someday people will start eating this. That's very nutritious. Okay, so I'm going to write it. Pollen kit is made up of, please write it with me because this you will miss. My bad with this space, carotenoids. And we cover almost all the functions of tapetum by doing this. Cool? Now there is a question asked in 2020, the year of easy paper. Vijay, you always need a break here. I'm kidding. What about ma'am? Later. Let me finish one topic here. We've just done what? Pollination. Which of the following is incorrect? Focus on the word incorrect. Answers in the chat box, please. You can say bye after the answer. Incorrect. For wind pollinated plants. Anybody? I'll give a break after the question. You're negotiating with me for a break. Huh? Guys, when you attend physics and chemistry continuously, do you guys do this? And those are actually very difficult subjects. I hope none of the teachers are listening, but bio is, especially botany, is one of the easiest subject in your entire scheme, syllabi. Don't you think so? I'm going to go and see and check with people. Did you ask for so many breaks in chemistry and physics? Huh? Now everybody is answering 2. Let me see. Which of the following is incorrect for wind pollinated plants? Well exposed or exerted stamen and stigma? Yes ma'am. Otherwise how will I throw and catch? Many ovuled in each ovary? No. We anyway have a scarcity of pollen grains. I don't want lot of styles and lot of platforms. Yeah, I will have one ovule waiting for all the pollen grains. So this itself is the answer, but let's explore. Flowers are small and not brightly colored. Ma'am told you it's an inflorescence, even the corn cob that you eat. It is the entire spathe inflorescence. What you're holding in your hand and eating, no? Nibbling on, that is the peduncle. Pollen grains are light and non-sticky. Makes sense to us? No problem. Sai, you are a darling. So you guys didn't take a break in chemistry, physics? Now comes artificial hybridization. What I'm planning? Listen to my plan. I don't carry a watch in my classes. It's going to be 10. We started at 8. Okay, so we'll meet at 10:15. What do you think? I didn't have my dinner, and I think energy is in your head. Anyway, 10:15 is a good idea. 10:15 is a good idea, guys. Let's meet at 10:15. 15 minutes. It's a 20 minutes break rather. Let me tell you, artificial hybridization human-controlled pollination and hence fertilization. When human beings got to know about Anenophily, Entomophily and all of this, they thought we are going to control it for our own greed. That's all. That's all. Meeting you at 10:15 sharp. Thank you. Eat food, drink water, use washrooms, but be attentive. Is it time? It's exactly the time. Yeah, without a watch. Alright people, please be back. This is the last segment of the class, I promise. Please call your friends, family, neighbors. I really mean it. See, the challenge that I have taken on this channel, and I say that in all the classes wherever I teach at any given point of time, is to make botany seem so palatable, if you will, irrespective of our preferences and interests. So I often tell people that, you know, get non-NEET aspirants into my class, yeah, and we will make everybody fall in love with botany. Second thing I always tell you guys, botany is in your kitchen. It's not in the textbook. I mean it is in the textbook. The syllabus is, the boundaries of the syllabus are, but your understanding of the subject is around you. Make sense little bit? So guys, if you have not shared this session with people you know, due to any reason, I think you should really do it. Now I want a lot of hearts in the chat box, fire in the chat box, like Gopika ma'am says, yeah, or whatever is your emoticon you're playing around with these days. Please, because the next segment of the chapter is post-fertilization events. Fertilization, double fertilization and post-fertilization. It is difficult to understand because it is not given very nicely in our revered book, which is NCERT. Yeah, and all the teachers, the entire team of Unacademy NEET English, my God, it warms my heart. So much of hard work everybody is putting only and only for you guys, isn't it? So teachers are standing for 7-8 hours, all the teachers for that matter, and it takes a huge team and lot of efforts to make every class possible. There's a lot of back-end team who works day and night on you folks, isn't it? Cool. So guys, one question I had, like I say, I tickled your brain regarding this one here. Why bees are not present around red flowers? Yeah. The answer to that question I expected you guys to give me. It's really it's an important fact in my head. They use UV radiation, and red under UV radiation will appear black, and they don't want to be around a black flower. So usually around red flower you will find butterflies mostly. Another thing I wanted to clear was, I think in Hydrophily we have written Vallisneria as hypohydrophily. Did we do that? Did we do that? Then you must know that Vallisneria is epihydrophily. Kindly correct it. Yes, I think I did that. Now, when human beings got to know that pollination is so schematic, they thought that if I choose which pollen grain, who will be the groom and who will be the bride, then I can get certain hybrid varieties with the chosen characters, which is intelligent. So artificial hybridization is abundantly used in crop improvement programs. In crop improvement programs. Now, I choose the male generation, that is the pollen grain, which will carry the superior characters as I see them. Superior character according to one horticulturist could be different. It could be the need of the hour, that in my particular astro geography, agroclimate. Yeah, agroclimate is the climatic conditions in that particular area. My crop needs, let's say, temperature resistance or light intensity resistance, or there could be a certain pest or disease that is attacking the crop, isn't it? So I choose the variety which is superior according to my need, and female, that is the stigma, is also chosen to get a hybrid which has the superiority. This is the basic scheme. Superior characters. Actually, in terms of genetics, we should say superior traits. Am I right or wrong? Character is not the right term. Superior traits. Is that clear? So now what's going to happen is, there are certain steps that I need to follow. Also understand, you know how genetics works. I would require a lot of generations of crossing to get a hybrid which has finally the culmination of the superior traits. Every cross will have these steps. First is emasculation. Given that your flower is bisexual, again you want to prevent any kind of pollination starting from autogamy, Xenogamy, or even Xenogamy, especially autogamy. So in a bisexual flower, removal of anthers. This question is coming repeatedly in your NEET exam, year after year. Once I remove the anther, I know that no kind of self-pollination is happening, but I want to prevent get-to-know or cross-pollination also. So I immediately bag butter or polythene paper. Okay, so I immediately cover both the male. You have chosen a male flower also. Yeah, you have removed the males here. This female, okay, this female and the male that you have chosen, the male flower, the anther that you have chosen, both have to be bagged. Both female and male counterparts. After that, once they attain sexual maturity, after sexual maturity is attained on both the sides, the actual crossing over is done. Crossing over, or in other words, simple terms, dusting chosen pollen grains on chosen stigma. And as soon as you do that, please bag them, because there are huge chances of contamination now. Okay, and you wait for, after that this process comes. Okay, so after bagging, you do this, and then you rebag them for the fertilization to happen, for the fruit to form, for the seed to form. You give them the time. and you rebag them. Once you have this seed in front of you, you see whether superior characters have been incorporated. This is what artificial hybridization is. If the flower is unisexual, this is the previous year question, not required, emasculation not required. In unisexual flowers, and you know why. There is no problem of self-pollination. Is that clear? Finally, we come to understand the natural process of fertilization. The entire course of events from landing of pollen grains till pollen tube enters ovule is called not fertilization. What is it called? Pollen pistil interaction. If this is successful, only and only if pollen pistil, if they agree to be coming together, then fertilization will happen. Now, what is the nature of this interaction? What is the nature of pollen pistil interaction? How are they checking their compatibility? I told you that Xine has compatibility proteins on the surface secreted by tapetum. That is the function of secretory tapetum. There is amoeboid tapetum, there is secretory tapetum. I know it's not in your syllabus. This is completely chemotropic. It is a chemical dialogue. And after the chemical acceptance of each other through recognition factors secreted from both the sides, the pollen grain germinates. The germination of pollen grains simply means the intine extends as a pollen tube and it is ready to travel through this. Okay. So now if this is a solid canal, sometimes it is solid, sometimes it is hollow. Then it is made up of pectin. It is made up of pectin. And the pollen tube. Pollen surface will have to secrete a lot of pectinases to be able to get transferred through this solid mass. And it has the permission to enter the ovule. This is the ovary. Inside the ovary you have the ovule, only through the micropilar end. Even if it gets an entry through other methods, can be through integuments as well, through the chalazal end as well, chalazogamy for example. It will ultimately have to curve and enter through the micropilar end. So, chemotropic is the word first of all. Secondly, what you need to remember is the pollen, when the pollen tube falls, let me draw it for you. Okay, this is the pollen grain. This pollen grain to start with was 3-celled. It had 2 male gametes and 1 vegetative cell. It has the exine, it has the intine, everything said and done. Now when it grows, okay, when it grows inside to finally reach the microbial end, it is only the tip of the pollen tube. It is only the tip of the pollen tube that grows. Okay, and at the tip, who leads the way is the vegetative cell or the tube cell. It will also lead the way, this is a previous year question, and it will be followed by the 2 male gametes, always. Some authors do believe that tube cell on this path goes away, disintegrates. It's like, you know what, everything is done, the final destination we have reached, we don't need to really be there. And some say that as soon as the pollen tube enters the microbial, the pore, that is the time tube cell degenerates because it wants to take the first collision with the synergid. You know, here we have the synergid and egg, and nobody is allowed to interact with the egg directly. Is this clear? Now, this phenomenon is called as siphonogamy. This is a very important term where only the tip of pollen tube grows in response to a chemical, okay, in response to concentration gradient. This is given in NCERT but not properly, you can add it there. Concentration gradient of calcium, boron, inositol. It's a complex. Sugar, boron, calcium. So if boron is of course it's not in your, it's a micronutrient. Calcium is still a macronutrient. Inositol complex. Is that clear to everybody? And because it moves through the passage of this style, this is called as intracellular, and because I am following the concentration gradient of a chemical, it is called as chemotropic movement. So all in all, everything is based on chemicals. Chemotropic movement. Now what happens when I reach? The fertilization happens, isn't it? Now before that, what we are going to do is, we are going to quickly understand what happens. Okay. So here you had the piriform outline, you had this was your egg apparatus present at the microbial end. Yes, this was the nucleus ready to take the collision of synergid. Do you all agree? Synergid. And here the queen is waiting with its N nucleus. Everything here, boys and girls, is N, N and N. Do you agree? Okay. So now I am telling you the very initial part of fertilization. Before we even arrive at double fertilization, we don't care. This micropilar end receives the pollen tube. Once again, at the tip tube nucleus, as soon as it reaches the micropilar end, goes off. Okay. So now what I have is, very easy, the two male gametes. And they have been guided by many things, obturator apparatus, and there are many things, but you know at your level it is filiform apparatus also, which was present with respect to synergid. Yeah? When these male gametes enter the egg apparatus through the micropyle, they interact with only one synergid. Only one synergid. Okay. So, let me continue here. This is our egg apparatus again. Egg is waiting. Yeah. Here you also have the central cell, the 2N central cell. Okay. I am just drawing the micropilar end of the embryo sac. Here you have the synergids. Good. Your pollen tube has got the permission to enter. You have once again the two male gametes. Yes. Now what I told you just now, this pollen tube carrying the very important constituent is going to either go to this one or this one. It is not allowed to interact with anything else. So let's say this is synergid A and this is synergid B, and pollen tube coming with certain force and velocity or whatever collides with this one here, the B. It is going to collide with the nucleus. The moment it damages the nucleus, this guy goes out into the thin air. It's completely gone, degenerates. Hence, this synergid comes to be known as degenerating synergid. Not given, but important. While the other one, who is very happy and celebrating that the accident didn't happen with me, waits here, and this guy here is called as the persistent synergid. Are we good? But that is a delusion. The moment this one degenerates, the moment this guy degenerates, let me see if I can. I knew it. Yeah, now this one is out of the picture. He sacrificed his life to take the collision for the queen. Then these two male gametes lie in the cytoplasm somewhere here, near the egg. They are allowed. And as soon as this happens, this one also degenerates. At this moment, mind you, antipodal cells also leave the scene, because now the actual meeting or marriage is going to happen. So out of the two male gametes, remember, one fuses with this is the male gamete, N, male, yes, and it has finally the egg. It finally has the egg. Female, but this is also N. Just think about this. We're not reaching anywhere. N and N is there. The problem is the second gamete says, where do I go? So now the ovule says, you know what, we don't have anybody. Antipodal cells have left the scene. Synergids are gone. The actual true marriage has happened. You guys go to, these guys were so notorious all their life, you guys go to the central cell. This is the central cell waiting, isn't it? That is why, my dear children, we call it double fertilization. We call it double fertilization. I have moved, yeah. So we are going to try to use this space and make the notes of it. Once the male gamete fuses with egg, it invariably leads to the formation of zygote, which is 2N, and this is called as nothing but true fertilization, because it is giving me finally the characters of both male and female, and I get a real plant with the zygote. There is mixing of characters that had to happen. But the other kind of fertilization, where the central cell fuses with male gamete, gives me a triploid tissue called as PEN, primary endosperm nucleus, which is triploid, and this is called as triple fusion or the pseudo fertilization. Is that clear? Somehow some patterns have come on this page. Yeah. So triple fusion, where 2N structure, central cell, but this topic is very easy to understand, and male gamete leads to a triploid tissue, yes, plus Xenogamy, which is a very generic term, you know, fusion between the N egg and the N male gamete is together leading to double fertilization, which is a phenomenon unique to only angiosperms. Okay, discovered by Novoshan. After this fertilization has happened, I have a zygote to take care of, which I am really interested in, because it is going to be my future plant, baby plant. But I also have a primary endosperm nucleus, which is triploid. So what happens is the question. Then we speak about post-fertilization event. Now what really happens is, due to the fusion of 3 nuclei along with the central cell, this pen is a very, very nutritive and active tissue owing to its ploidy. When I say active tissue, understand the meaning. Owing to its ploidy, it has a power or ability to derive nutrition from the treasure of nucellus. All of this is happening inside the ovule, na baba. All of this is happening inside the ovule. Nucellus is still there, having all the nutrients, everything. So ultimately, when pen develops, it becomes nucleus will finally become PEC, primary endosperm cell. And this primary endosperm cell, when it multiplies and develops, it will form a tissue called as the endosperm. And endosperm development has to precede zygote development, because otherwise zygote cannot derive nutrition from the parent, from the mother's womb. It is the endosperm which will nurture or mother the developing zygote. We understand it is important, but we need this food laden nutritive tissue to proceed, to take the lead. Is that clear? Now how does it happen? Endosperm development is a very, very easy thing. You should know that this is the first event, there is no breaking the sequence. Embryo development will automatically happen. When the ovule has its proper endosperm, it will get a message to start the embryogeny. And after the embryo has formed, we call the ovule to have conceived. So that ovule which has the developed dicot or monocot embryo according to the genetics, whether I am a pea plant or I am a wheat plant, then ovule develops itself into seed. So these are markers of the events, Baba. It's not happening randomly. Once this happens, it's a cue for the next step to happen. This is the third step. And as ovule develops into the seed, the ovary which is carrying the seed undergoes differentiating changes to become the fruit. Okay, become the fruit. Now when I speak about endosperm development, there are various types. It's altogether a section in this chapter. But at your level, what you have to understand is the simplest and the commonest type, which is the nuclear type. As the name suggests, this endosperm is just undergoing repeated free nuclear division. The key word is free nuclear division. You can see it, right? So the key word is free nuclear divisions without cytokinesis. Later stages will have cytokinesis. Do you see that? Which will start from the periphery. NCERT writes this line, I am just telling you, I am decoding it. There you see, from the periphery to the center. So no structure wants to be just like that, free nuclear, multinucleated structure. It tries to, like it happened in formation, organization of embryo sac also. So later stages it might see, if not completely a cellular. This type of endosperm, boys and girls, is the most common one. And the example that examiner wants you to know particularly is the water of coconut. I can hear all of you screaming. That is the only thing you know in this chapter. Now, another variant and less common one is the cellular one. The cellular endosperm is simply proper division, karyokinesis followed by cytokinesis. So wall formation happens along with nuclear division. Happy? Now classic example is the kernel or the meal. Of coconut. So what we are eating in coconut is essentially, that is why coconut is said to be so nutritive. It's considered as a perfect food because it has all the nutrients which endosperm has built in while it was changing its stages from pen to peck to becoming a full-fledged endosperm tissue. There is a third type which is not at all asked since last 10 years, if I can say, halobial endosperm. It's a sort of a mix between nuclear and cellular, and it also grows up to be nuclear. Yeah, it is seen in halobiales order, not given in NCERT, not needed, I assure you. Now what is important is embryogeny. Yeah, the problem with embryogeny is it is there in your syllabus, but NCERT has not done a fair job. I am going to mention the questions that come from this topic, the exact keywords I am going to give you, but I cannot just mention the keywords. It is not right without explaining the process. But I also promise I am going to make it super easy for you. Ready for that, everybody? Let's give it a try. Now, good news is, I am going to start with the good news. Not that there is a bad news in store. Dicots and monocots have very similar embryogeny, monocots being simpler. So you don't have to, I am not going to worry you with monocot embryogeny at all. Good? Makes sense? After embryo formation, you've already studied the structure of dicot seed. You know how embryo is, cotyledons, what is plumule, what is radicle. So this topic is actually very easy. Now here is your zygote. And where is zygote formed? Remember, you can never forget the polarity. We are still in the polarity. So this is the micropilar end. Okay, let's make it, yeah, this is the chalazal end. Good. This zygote will now, with the help of all the support and nutrition and mother-like care from endosperm, is going to elongate every cell which is ready to divide. But here also, as the plant likes to create some different scenarios, unequal mitosis will happen. So the first division itself is an unequal mitosis, and as a result of which, needless to say, you are going to get a smaller cell and you are going to get a larger cell. Is that very clear for everybody? Yeah. The smaller cell is going to be called as the embryo cell, or simply the terminal cell. Is that clear? And this larger cell is going to be called as the basal cell or the suspensor cell. Is that clear? Now, as the name suggests, this suspensor cell is going to form the suspensor, and embryo cell is going to form the actual embryo. So it is not the entire part of zygote. I want to check the orientation, whether M and C are correct. Yeah, it is absolutely correct. Okay. So because examiner, if again it's a rainy day, we have to, on this channel, you are not here to just complete the syllabus and do what others are doing, right? So we have to be prepared for tough questions as well. NCERT is speaking about these points but not mentioning it very properly. So remember, he might ask you, he is going to ask you which end from the orientation, which end, is it the micropilar end or it is the chalazal end, will have the suspensor cell. Now what is going to happen is, although the further divisions happen simultaneously. Please hear me out. The further mitotic divisions will happen simultaneously in both the embryo cell and suspensor cell. Starts more in suspensor cell, but I am going to take one at a time. So what I do is, I am going to draw the embryo cell as it is. Okay, and this suspensor cell is now going to undergo transverse divisions. Told you in the beginning, this evening, early evening, move your scalpel, move your knife or scalpel, transverse. Yes? So now what you are going to get is a structure like this. I have made 6 celled structure. This 6 to 8 celled structure is called suspensor. Now boys and girls, all of this is happening where? Inside the ovule. This is surrounded by nucellus. The zygote has 2 points of nourishment and it is intelligent. At this end, it has the endosperm lying for it. But it is also surrounded by the big magnanimous nucellus or body of the ovule, which is starting to get consumed. Okay, makes sense? So when you get the final embryo in a seed, you have studied in class 11th, you have endospermic seeds, you have non-endospermic seeds. What does that really mean? Either if I am a dicot, you know what is going to happen? I am going to consume endosperm. I don't care. I have 2 cotyledons to develop. I have a lot to do. But the nucellus in both dicots and monocots gets consumed. One of the major reasons for that is, let's erase this cell, okay, and tell it to become like a seat, like somebody who's going to suck the entire nucellus. And the botanical term for that is can you appreciate? Oh yeah, it is Haustorium. Well, this is nothing. This is nothing but the last cell of suspensor. If I am 10 celled suspensor, it is the 10th cell. If I am 6 celled suspensor, this cell gets swollen. You remember hostorial roots in Cuscuta, Amarbale? We spoke about in the Avengers batch just now, day before, when we spoke about parasitism, holoparasites, hemiparasites. Rings a bell? Yes? So hostorium simply means it's going to suck nutrition. Now let's talk about this cell. Remember, while this is happening and I am talking to you, this cell is not waiting. It is quickly going to change into embryo because that's the real deal. But I am teaching you separately so that you understand. Now this cell, which is adjacent to this embryo cell, is going to change. It's not going to become very swollen, but this cell is called as hypophysis. Okay ma'am, as you say, in the life of the plant, whenever I come across hypo, it is usually to do with the underground portion, the descending part of the plant. Whenever I have come across the word epi, since we are going to talk about the seed formation, right? So you are getting the idea. It is for the ascending portions of the plant. With me? So this guy here is not going to do much but aid a little bit of. Let's write it. Yeah? What is he going to do? Take part in root cap, some amount of radical also. Take part, not do it entirely. Take part in, because that is the job of this embryo cell. Take part in root cap formation. Good, no problem happening. This is the deal of the larger cell or the suspensor cell. Anybody who has a doubt? Up until here. Very important topic, guys. Can't take it lightly. Oh God. All good? Somebody is nice enough. Please, I have to have space. Okay, no problem. Now let's see what happens to the embryo cell. Let's isolate that guy. This embryo cell which is being pushed by the suspensor towards the endosperm and also getting nutrition from nucellus is very comfortable now to undergo its first vertical division. There is nothing of the sort unequal. It is a mitosis. If somebody doesn't mention it specially, it is a mitotic division. Whenever there is growth and development in any body, it will be mitosis. Whenever there is reproduction in any body, there will be meiosis. Please don't get confused at these small things, small matters. Make your mistakes worthwhile. Like I didn't know an example. Like I didn't know hypohydrophily or able. But as far as cell division is concerned, let's not do that to ourselves if we are claiming to be NEET aspirants. So now imagine there is a circular structure. You have a onion, you have an orange. Make a vertical. Scalpel, knife, vertical, vertical, vertical. So what am I going to get? I am going to get this. Ladies and gentlemen, this first stage is called as the pro-embryo. That is where NCERT is not doing a good job. It is speaking of the various stages, but we don't know how we got there. Okay? So even if you have to watch this part of the session again while reading NCERT, give yourself that chance and service. Okay? Now what will happen? It will undergo another division. You know how plant is. It loves multiple mitotic division. The second division, interestingly, is also a vertical division. But if you cut you can't cut the same structure like this. So scalpel, I've cut it like that. I have two cells. Now the second division has to be vertical, perpendicular to the first vertical division. So it is vertical in this plane. Try to make sense. It is okay. So what do I get if I cut it here? I get I get 4 cells. Happy? This cell, once again, eyes on the screen. This is vertical division number 1. This nucleus is going to undergo vertical division number 2. This one is going to undergo vertical division number 2. This is what you get. This is called as the quadrant stage. Are we on the same page, people? Moving further, the embryo cell is still not happy. You know, he's like, even before I was born, my parents, male and female generations, they always had multiple mitotic divisions. I want more cells. So the third division takes place. Third transverse division takes place. Good. Now imagine, you have to imagine this. Unless you go to the kitchen, but there's your cutting technique, your dexterity should be good. Don't try this on your own without any supervision, because I have cut my hand, okay? So, but if you can really take the help of your imagination, you made a cut like this, then this, and now you're going to cut these two further transversely, these two further transversely. Eyes on the screen, makes more sense. Transverse for both. So 4 cells at the top, eyes on the screen, and you get 4 cells at the bottom in 2 tiers, in 2 compartments. Once again, people, once again, this is what you had to start with in the quadrant stage. Now you take your knife, and you don't have any care in the world, because it cannot be another vertical division, ask me. You move it transversely. So forget about this, move transversely your knife here. You get 4 cells, okay, happy. Forget about this, move your knife here, you get 4 cells. But now you have 8 cells. You have 8 cells distributed in 2 compartments. A 1 and 2 and 3 and 4. Yes, because don't get scared, I am here. This was, forget about this outer tier. This was already present. Now you have divided these two, so the additional cells, additional nuclei have come outside. Similarly, you always had this. You always had this. And now when you move the knife, you get 2 additional cells. And a total of 8 nuclei or cells. This is called as, this, my dear children, is called as the octant stage. These stages are of paramount importance. If you are thinking that I am just giving you some terminology, please open up previous year papers and help yourself to understand that how questions are asked. Are we good? Now, This upper tier is called as epibasal. And this lower tier is called as the hypobasal. This hypobasal cell, I told you, will make the descending organs. Radicle, root cap. Okay, so this is going to aid in the root formation. Which will include radicle, root cap. And epibasal cell, a previous year question is which layer of cells will form cotyledons along with the plumule. So, cotyledons as well as plumule is formed by epibasal cells. Now children, as this structure is growing further, it hasn't stopped dividing yet. After octant stage, what you get in life is another stage which is mentioned as the globular embryo. Some authors do believe that octant stage is the globular embryo because it's like a globe, okay? But I would say it develops further and becomes a huge rounded structure, and then the embryo cell realizes, oh God, I need to make 2 cotyledons. What do I do? So now it has to develop a constriction. It has to develop a constriction here. Yes, epibasal, plumule. And as it begins to do that, it sort of contracts itself and becomes a triangular. This I can say is not in the syllabus. But up until here, starting from proembryo, quadrant, octant, globular, everything has to be done. Yes, this is not mentioned, but then why are you studying about embryogeny if you don't want to know how embryo is formed? Then what is embryogeny? After that, finally, when I have the proper embryonal axis, which is going to deepen further and give me the 2 cotyledons and the whole orientation of epicotyle and hypocotyle, which we are going to see. This stage is the most asked stage in this, called as the heart-shaped embryo. This transitions, after that you get the proper embryo that you see in morphology also. Okay? So, happy? Healthy? Proembryo, let's revise this. First stage is proembryo. Second is quadrant stage. You should also know, because it is so interesting, which division is taking place where. And actually you should be making the mind map. Because you know what? The admit card is going to have your name. And then comes the octant stage. And then is the globular stage. There are many stages actually. But like I said, I promised I'm going to make it easy for you. And finally, the heart sheet. Good? Somebody? Thank you. Now what will happen? This will happen. I have not labeled the parts because this is a previous year question. So what I have done is I have taken the image of the question, wherein these questions are on high rise these days. The examiner is giving diagrammatic questions left, right and center and asking you to label the parts. So this you already know. What will happen here? What is this? What is this? So now imagine a embryonal axis, yeah, passing through this point. Then this is the plumule. Is this visible? No. So A is plumule, isn't it? This descending region is the hypocotyle. So C is going to be radical because hypocotyle gives rise to embryonic root. Are we clear on that? And these are the two cotyledons. Simple. Yeah? Below the level of attachment of cotyledons, you have the hypocotyle region. And above that, you have the embryonal axis is also sometimes called tigellum. All of this we'll do in morphology as well. Good. Now we are going to go to the monocots. I promise I'm not going to worry you or burden you with monocot embryogeny because there is nothing to see except a point which should arise from common sense. People, let's focus on the fact that after globular embryo, when embryo realized that it has to form two cotyledons, that is how it got triangular stage and heart-shaped stage. Monocot embryo, monocot seed is a single cotyledon. Hence, I am not going to see these stages, but I am definitely going to see all of this. The epibasal cells, this and this and this, if it is not clear, this and this. Okay. So, transverse divisions are happening in two parts. Like I said, don't try it unless you are destined to be a surgeon or a dentist. Then you will naturally have certain finesse or precision in your hands. Okay. So now what we are going to do is we are not going to write the entire process again. The suspensor in monocots is hardly 2 to 3 celled. It is not 6 celled. So the major activity in embryogeny that you see is at the level of embryo cell. Okay? And the finally developed embryo is this. So no stages after globular embryo, suspensor cell, suspensor is 2 to 3 celled. Now this could be the reason that monocot seed told endosperm, you know, your suspensor didn't push me only, I didn't get enough nutrition, please help me till the end. So when we get the monocot seed in our hand, most of them except orchids are carrying the endosperm. Yeah, you should know the exceptions. So now if you see this single cotyledon is shield shaped, called as scutellum. Isn't it? This is your plumule. Sorry, this will be your, in monocots we have added sheath. We have the coleorhiza, coleotile. Yes, this will be the plumule. What will be the plumule? Here, embryonic shoot. Plumule is what? Embryonic shoot. Going to form the shoot apex later. Now, do you see this here? One of the favorite questions. This is appreciated in grasses. Reduced second cotyledon, and that is called as epiblast. So this spiny structure that you see here in your textbooks, also same diagram is there. I have taken NCERT diagram. Now we come to the hypocotyle region, descending parts. This will be your radicle, just like plumule. Okay, and this is the radicle. This has to be the root cap. And this, my dear children, has to be the extra sheath present in monocots to protect the root and the plumule. In case of root, we call it coleorhiza. Okay, this is the So I want you to look at all the diagrams, every diagram of NCERT at least 100 times. Oh, 100 is a great number, that's why ma'am said it. No, count on your fingers. 100. And look at the labelings, because nobody will ask you to draw in the exam. So even if you think, you know, I'm wasting a lot of time drawing, although drawing a diagram again and again helps in better retention, but I don't want you to be finicky. I don't want you to hold on to the fact, oh, my diagram is not neat, my diagram is not coming out well. Make this practice that you are revising the labels every time you see the structure. Now comes apomixis and polyembryony. This is the last part of the chapter. Is everybody okay? Again, apomixis and polyembryony is repeated question year after year. Ready? If you guys are ready, just give me a fire sign in the chat box. Everybody. Now, apo means absent. Mixing here simply means fusion or fertilization. Okay? Fusion or fertilization. So, formation of seed without gametic fusion is called apomixis. Okay, thank you. Now, it resembles, this is a conceptual question or AR question. It resembles sexual reproduction as you get the product of the sexual reproduction. You get a proper zygote embryo, you get the seed. So, resembles sexual reproduction in terms of seed formation and asexual reproduction. Can you tell me how it resembles asexual reproduction? Thank you, Laharika. Oh, Laharika is still up, huh? In absence of what? The main act of sexual reproduction. What is the main act of sexual reproduction, guys? Fertilization. No problem, ma'am. Easily done. Is this clear? Now, it is seen in both the dicot and monocot advanced families. That is the beautiful part. Which is the advanced family in monocots? Come on, which is the advanced family in dicots and monocots? So, quickly, it will be seen in, I have said about this family many times, Astraceae family, which is in your syllabus, and the family of grasses, Poaceae family. This is one intelligent way of the plant to retain purity of its characters and also to sometimes prevent. The plant does it on its own. It's not, of course, human beings have replicated it for their own good, but this saves a lot of time that is wasted during sexual reproduction. Okay, and it maintains the purity of, and you're getting the seed. So, as a horticulturist, you are very happy. Now, type 1 Apomixis is called as let's do recurrent agamospermy first. Now, your NCERT doesn't speak about the types, and trust me, there are as many classifications of Apomixis available in different textbooks as are the families. So, what we are going to do is, I am going to give you the types or the classification which will go in tune or align with NCERT examples. So, for example, in recurrent agamospermy, again A is absent, gamo is fusion, spermy is coming together of gametes. Spermy is actually seed. So, seed formation is happening without any fusion of gametes. Why does this happen? When any cell of embryo sac, listen to me carefully, I am going to make it very easy as is the promise. Zygote was coming as a result of fusion of haploid egg and haploid male. I don't have the male. I have only one parent. So, now you are getting the zygote or the seed from any cell of the embryo sac. Any cell of embryo sac gives rise to zygote. Now the problem is zygote is 2n. Oh my god, zygote is 2n. An embryo sac was told to us as a completely gametophytic generation. Well, the catch in the story is this is happening because embryo sac develops directly from megaspore mother cell. And what is the ploidy of megaspore mother cell? We studied early evening. This was the concept, ploidy and mother cells. Or nucellus. What is nucellus? The body of ovule, sporophytic generation. Makes sense to all of us? I am just going to make it very easy. Is this difficult? Then I'll stop. To just keep you safe and healthy, is a type of apomixis, then it's a different kind of complicated. I would recommend that you learn this. And if nucellus is taking part, then that is simply apospory. But ultimately, the result is the same. Plants like to break rules. Okay? And then comes the second one, which is rather given in NCERT. Which is Adventive Embrony. And actually, polyembryony that you keep studying. Vijay, it's done. This is the last topic if you're thinking. It leads to polyembryony. How? There is an egg here. Okay? It is N. It is happy. It is waiting for the male gamete to come. That male gamete will enter and I will become a zygote. That is possible. That will make a viable zygote. But sometimes the surrounding integument will become cancer-like, and these cells of the integument will start proliferating. So much so that, and see, integument is a part of the sporophytic generation. It's a diploid structure, agreed ma'am. Now when it penetrates the embryo sac, I do have a proper and true zygote. I might have a proper and true zygote. There are no rules to that, which was formed by the fusion. But when this happens, that all the cells of the integument grow and penetrate the embryo sac, what they form are multiple embryos. And that, ladies and gentlemen, it was discovered in citrus, which has 2 to 40 embryos. out of which one will be the true one, like I said. The other are adventive. The consequence, what you just saw, is polyembryony. Poly means multiple. Good. It is also seen in onion. Genus Allium will also show it. Onion and citrus are the two examples that you have in your textbook. Mango is also mentioned by the textbook. So might as well mention it. So polyembryony. Now I'm going to write polyembryony is seen in these three. You might say, ma'am, you can simply write adventive embryony. No, that's the thing. That's the thing. Polyembryony. So I'm still telling you extra than the textbook because I don't want you guys to be lost. But you have to understand this is a huge and dense topic. Just so that you understand polyembryony, I told you adventive embryony. So the classic examples are citrus, mango and onion. Good. So that is how it looks. See, this is the integument that became proliferative, uncontrolled divisions, and now it is going to penetrate into the embryo sac. Polyembryony. Is that clear, everybody? So this finishes the chapter of sexual reproduction in flowering plants. Tomorrow, the link is given in the description box of the session. You should like this session, subscribe to this channel if you haven't subscribed, and you should share this session if you haven't shared. Please join the official Telegram group of Unacademy NEET English channel. Please join my Telegram group. The link is given in the description box. If not, you can join at Pankhuri 007. All the teachers, everybody's link is given. Please, this team is going to be reckless in your preparation. All of us teachers are going to go and make sure that every NEET aspirant is counted for and you get the best preparation. Yeah, that is what Game of NEET stands for. There's Avenger 3.0 going on. You can use the code PM1 live on Unacademy profile on Unacademy app for the special class tomorrow, and I will see you next time. Thank you very much. Good night.